Namaskara Karnataka. Welcome all of you to Vedantu Karnataka. Hegidira ellaru chenagidira. So yesterday was one big revolutionary day because 18 years wait admele finally RCB has become the champions. That is, they have the trophy in their hand. Alva. So young RCB finally after 18 years they waited. No, I want all of you also to learn this very big lesson that hard work, consistency, not giving up can make us reach somewhere, and that success is going to be more merrier than any other success. Nanne nodbodu entire Bangalore was celebrating RCB. Ivattu kuda the entire rally ella nadita ide illi Kaban Park hatra. So ಆ ಥರ ಒಂದು ಸೆಲೆಬ್ರೇಷನ್ ನನಗೆ ಡೌಟು, ಲೈಕ್ ಇಫ್ ಎನಿ ಒನ್ ಎಲ್ಸ್ ಹ್ಯಾವ್ ಗಾಟ್ ಇಟ್, ದಟ್ ಇಸ್ ಬಿಕಾಸ್ ದೇ ಹ್ಯಾವ್ ಒನ್ ಆಫ್ಟರ್ ಸೋ ಲಾಂಗ್, ದ ವೇಟ್ ಇಸ್ ಸೋ ಲಾಂಗ್ ಅಲ್ವಾ. ಸೊ ಆ ಥರನೇ ನಿಮ್ಮ ಎಂಟೈರ್ ಕಾಲೇಜ್ ಲೈಫ್, ಸ್ಕೂಲ್ ಲೈಫ್ ಎಲ್ಲ ತುಂಬ ಲಾಂಗ್ ಆಗಿರುತ್ತೆ. ಬಟ್ ಆಫ್ಟರ್ ದ್ಯಾಟ್ ವೆನ್ ಯು ಬಿಕಮ್ ಸಕ್ಸಸ್ಫುಲ್ ಇಟ್ ವಿಲ್ ಆಲ್ ಟರ್ನ್ ಔಟ್ ಟು ಬಿ ದ ಬೆಸ್ಟ್. ಓಕೆ, ಸೊ ಆ ಒಂದು ಪಾಸಿಟಿವ್ ನೋಟಲ್ಲಿ ಇವಾಗ ಕ್ಲಾಸ್ ಸ್ಟಾರ್ಟ್ ಮಾಡೋಣ. ಸ್ಟೂಡೆಂಟ್ಸ್, ಪ್ಲೀಸ್ ಲೆಟ್ ಮೀ ನೋ ಇಫ್ ಐ ಆಮ್ ಆಡಿಬಲ್ ಆ್ಯಂಡ್ ವಿಸಿಬಲ್ ಟು ಆಲ್ ಆಫ್ ಯು. ಎಸ್, ಕ್ವಿಕ್ಲಿ ಲೆಟ್ ಮೀ ಆಲ್ಸೋ ಚೆಕ್. ಬಿಕಾಸ್ ದೇ ಹ್ಯಾವ್ ಒನ್ ಆಫ್ಟರ್ ಸೋ ಲಾಂಗ್. Yes, perfectly audible and visible. Hi. Yes, quickly let's mark the attendance as well. ಬೇಗ ವಿಡಿಯೋನ ಲೈಕ್ ಮಾಡಿ, ಓಕೆ. ಆಮೇಲೆ ಫ್ರೆಂಡ್ಸ್ ಜೊತೆ ಶೇರ್ ಮಾಡಿ. ನಿಮ್ಮ ಕಾಲೇಜ್ ಇರೋ ಫ್ರೆಂಡ್ಸಿಗೆ ಈ ವಿಡಿಯೋ ತುಂಬ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಬಿಕಾಸ್ ಕಾಲೇಜಲ್ಲಿ ನೀವು mostly you will skip this chapter because you think that this chapter is very difficult. So Gopika Ma'am is going to make sure that this chapter is going to be super easy for all of you. I will make sure that this chapter becomes very simple, and you know, I will break it down as much as possible. Okay, start madana students. Ready na? Full energy those students. The students. Okay, RCB has won. Okay, life has become like there's so much hopes in life. Okay, so why you people not having hopes? Come on. So we are going to do molecular basis of inheritance part 1 ivattu. Nale part 2 madana. So I will be completing the chapter in 3 or 4 parts. Max to max 3 parts al complete madbodu ee chapter na. So ee chapter alli ella in depth amele listen to me carefully. After this class you do not have to read your NCERT because I have also put the NCERT snips. Okay, so NCERT snip add madidini so that you don't have to struggle. Okay, you don't have to struggle with, you know, reading NCERT as well. Okay, very nice, Uday. That is the energy I want till the end of the session, and I want all of you to share the video with all your friends. Okay, chalo start madana students. So first, many students were asking me, ma'am, namigilli Vedantu learning center idya ilva anta. Vedantu learning center ide students. Ellide Vedantu learning center? JP Nagar alli ide. Okay, that is in Bangalore. No, so those students who are in iden connect ಆಗ್ತಾ ಇಲ್ವಾ? ಓಹೋ ಒಂದು ನಿಮಿಷ, ಬೇರೆ ಪೆನ್ ಟ್ರೈ ಮಾಡ್ತೀನಿ. So those students who are in JP Nagar, okay, JP Nagar can come and visit the VLC center that is in Bangalore. Okay, Bangalore. So if you are looking for NEET, KCET, illandre JE coaching ke, if you want to come and check out, you can come and check out. Okay, and there is a link in the description. So our link in the niಮಗೆ demo, like if you want to book a demo, illandre on book a visit. If you want to see the VLC centers, you want to see the, you know, how is the setup, everything, how are the teachers there, then you can definitely book a visit. Okay, clear to all of you? Very nice, very nice. Okay, hello, hi, bye, you can tell later. Now we will start the class. I want all of you to listen carefully to the class, and all these notes will be available on Vedantu Karnataka Telegram channel. Clear? Okay, now introduction. Students, normally what happens whenever we see a baby, just one baby, we tend to tell that, oh, you look exactly, the baby look exactly like the father, the baby looks exactly like the mother. Yeng ishtu easy agi helbodu because we know that genetics anta on term ide, andre genes, there is something called as genes, and these genes are inheriting those characters alva. If physical traits agirli, so physical traits ella color of the eyes, height, tell very easily, oh, the baby looks exactly like the father, the nose is exactly like the mother alva. But who is the reason behind this alva? Who is the reason behind this? We know it's a small portion in the chromosome, but what makes this chromosome? Who is going to carry all this genetic information? Is there a cell who is going to carry all of this genetic information? Gottirbeku. So I'm going to introduce you to something called as nucleic acid. So students, before I tell nucleic acid, understand a cell, okay, that holds all informations. Okay, a cell that holds all informations. Okay, that is what we are going to talk about. My handwriting is not this bad, the board is very bad, I feel. Okay, so yeah, a cell that holds all the information. And now when I talk about all the information, you should remember that these people are going to have, okay, there is something called as nucleic acid, and nucleic acid is of two types. But first students, artha madkoli, nucleic acid was found by a person called as Frederick oh, nice. Try madana. 1 second. Me figuring out the board. Okay. Frederick Misher. Okay. So Frederick Misher was a person. Okay. Frederick neither anta bantu. Okay. Never mind. Namak first in the bariyana. So yeah, there was a person called as Frederick Misher. Okay, what was his name? Frederick Misher. What was he doing? He was actually working on a different cell. Okay, he was working on some cell, students, but ಏನಾಯಿತು by ಇವಾಗ ನೀವು ಮನೆಯಲ್ಲಿ ಹೋಗ್ತಿರಲ್ವಾ, so your mom will tell ಏ ಪುದೀನ ಎತ್ಕೊಂಬ ಇಲ್ಲಾಂದ್ರೆ ಕೊತ್ತಂಬರಿ ಎತ್ಕೊಂಬ, they she'll tell. Okay, what you will do, you will go and you will open some random box and luckily you will get off pudina and you will be like, yes, success, because ನಮ್ಮ fridge ಅಲ್ಲಿ ಒಂದು 100 or 50-60 Amul boxes irutte, illandre biryani boxes irutte. So we do not have proper, like ee foreigners ella iktaralva aa tara ondu proper tumbler nam maneyalli iralla. Usually ad bandbitu almost this plastic, okay, whatever biryani containers, ice cream containers ella irutte. So by mistake you are going to take this one thing and you are like, oh please let it be pudina, and you open and it is pudina. So it's like by fluke, right? By fluke. Illandre on multiple choice question alli A you wanted to answer, but in your mind it's like it's going to be B, and you answered B and you got it right. So by fluke anta heltivi alva. So Frederick Misher also by fluke he found out something called as nucleic acid. Okay, he found out something called as nucleic acid, and that was the turning point, because he was able to tell that this cell has all the information that is required, right? This cell has all the information that is required. Now students, nucleic acid are of two types. Okay, two types you are going to study in this chapter. First one is going to be DNA. Second one is going to be RNA. DNA full form eno? deoxyribo nucleic acid. Deoxyribonucleic acid. Correct? Yes or no? Now, RNA means what? Ribonucleic acid. Ribonucleic acid. Alva. What is the difference you see in their spelling, students? You will notice that, ma'am, here you mentioned something called as deoxy. So, students, see deoxy and one oxygen is less compared to this, right? So, absence, right now I am only writing absence of oxygen. Okay, if you want, I'll write the whole thing. Absence of oxygen, but I'll nodi ribonucleic acid. So that means here we definitely have a presence. Illi en absent ide adil present agide. So presence of oxygen. Correct? Yes, ma'am, correct. So this is the biggest difference between both of them. Deoxyribonucleic acid means absence of oxygen. Ribonucleic acid means presence of oxygen. Now, understand, students, e nucleic acid bandbittu, what is their main job? They are going to have all the information, right? They are going to have all the information that has to be that has to be inherited. Alva, that has to be inherited. So introduction bandbittu, Frederick Misher found nucleic acid. Now nucleic acid alla Frederick Misher actually found nuclein. Why? I'll tell you. First I told you nucleic acid. Why? I'll tell you. See, he found something called as nuclein. Nuclein, what was it? No one knew. Then another person called Altman came and told that actually this nuclein has an acidic property. It has an acidic property. So why don't we call this nuclein as nucleic acid? But see, nim textbook alli Frederick Misher found nucleic acid anta directly bardidare. So that's why even I told you people in the same way, okay, but extra information really. So it was first called nuclein, but Altman, another person, found that there was an acidic property in this, because of which we can call it nucleic acid, okay, because of which we can call it nucleic acid, okay, clear to all of you? Now, this, when I tell a cell that holds all the information, students, one thing is assured, that the cell should have extreme accuracy, correct? Accuracy should be there, otherwise there is chances that mistakes can happen, and mistakes andre now exam al bariyo on the answer mistake tara alla, on complete mutation can happen, one genetic syndrome can happen, alva, it can be life threatening. So properly the genes have to be accurate, that is while copying, while converting, everything has to be proper, right? Everything has to be proper, understood? So look here all of you, let me also show you NCERT. So what is NCERT's introduction? Let's see. Okay. Deoxyribonucleic acid and ribonucleic acid are two types of nucleic acid found in living system. DNA acts as a genetic material, okay. DNA acts as a genetic material in most of the organisms. So, illi ಏನು ಹೇಳ್ತಾ ಇದ್ದಾರೆ ನೋಡಿ students, illi when you compare deoxy and ribonucleic acid, you will be like, ma'am, absence of oxygen would definitely be a disadvantage. But DNA case alli adondu advantage. Why? Because DNA becomes more stable. So, because of which, students, this DNA is a genetic material for almost 99% of the organisms. Okay? Or we can tell majority of the organisms, okay. On the other side, RNA is only genetic material for few viruses, okay, few viruses like your TMV, okay, HIV, okay, retroviruses. So these are a genetic material for only few viruses, but on the other side your DNA is a genetic material for most of the organisms, okay. Chemistry class is happening every day in the channel. I don't know why you people can't see the chemistry class, okay. It's every day happening on the channel, you people are not attending. DNA acts as a genetic material in most of the organisms, and RNA, okay, RNA, though it also acts as a genetic material in some viruses, mostly it is found as a messenger, okay, tumba important. RNA, even though they are not genetic material for all the organisms, they are found to be as messenger, okay, messenger. Now, RNA has additional roles as well. It functions as adapter, which you will study again later in the translation chapter, translation topic. Structural, and in some cases act as catalytic molecules, okay. Catalyst andre someone that increases the rate of reaction but does not directly involve in the reaction, okay, involve in the reaction. Clear to all of you, yes or no? Okay, very good. Now, class 11th, you would have already studied the structure of nucleotides. Students, where have you studied the structure of nucleotides? You would have studied it in biomolecules chapter. How many of you remember? How many of you remember your 11th topic? You would have studied this in biomolecules, where you will be studying about different types of purines, pyrimidines. So, other than salpa continuation ee chapter li ide, okay na? So, nucleotides and the way these monomers units are linked to form the nucleic acid polymers, okay. Now, we are going to understand the further. See, here this paragraph is the most important, students. See, paragraph alli ella words important because ಸ್ವಲ್ಪ numbers ಕೊಟ್ಟಿದ್ದಾರೆ, so it can come as match the following. ನೋಡಿ ಇಲ್ಲಿ, DNA is a long polymer of deoxyribonucleotides. I will teach you all of that, don't worry. The length of the DNA is usually defined as the number of nucleotide or a pair of nucleotide referred to as base pairs present in it. This also is a characteristic of an organism. For example, a bacteriophage known as 5174 has 5386 nucleotides, okay. In number you have to remember and keep, okay. Next one, bacteriophage lamla has 4850502 base pairs. Illi nodi difference, students. Id banbitu, okay, this is a nucleotide. Alva, this is a nucleotide. That means what? There is no base pairing, okay. There is no base pairing, okay. No base pairing andre eno, this will be a single strand, okay. It's going to be a single strand. SS anta bardidini, that means it is a single strand. Clear? Now, here this has a base pair, so obviously this is going to be DS, double strand, okay. DS andre double strand. Next one nodi, E. coli, okay. E. coli, that is E. coli has 4.6 into 10 to the power ಸೊ ಸ್ಟೂಡೆಂಟ್ಸ್ ಅವರು ನೋಡಿ ಇದು ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. ಸಡನ್ಲಿ ಇಫ್ ದೇ ವಾಂಟ್ ಯು ಟು ಡೂ ಡಿಪ್ಲಾಯ್ಡ್, ಜಸ್ಟ್ ಡೂ ಇಟ್ ವಿತ್ 2. ಓಕೆ, if they ask you diploid, you just do into 2. Okay, into 2, so it will be 6.6. clear? Into 2 you can do. Okay, now polymer ಬಗ್ಗೆ deoxynucleotide ಬಗ್ಗೆ ಮಾತಾಡ್ತಾ ಇದ್ದಾರೆ. So let's see what those things are. Okay, so structure of a polynucleotide chain. So students, remember, very simple. Nam DNA nucleic acid anta you already understood. Now what is this structure made up of, right? These structures are made up of something called as nucleotide, which you would have studied in your biomolecules chapter. Okay, nucleotides. Now what does this nucleotides have? Nucleotides are made up of 3 components. Nucleotides are made up of 3 components. Okay, ಸ್ಟೂಡೆಂಟ್ಸ್ ಪ್ರಾಪರ್ ನೋಟ್ಸ್ ಬೇಕಂದರೆ ನನ್ನ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲಲ್ಲಿ ಜಾಯಿನ್ ಮಾಡ್ಬೋದು. ಸೊ ಅಲ್ಲಿ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲಲ್ಲಿ ನನ್ನ ಹೆಸರು ಹಾಕಿದ್ರೆ ನಿಮಗೆ ನನ್ನ ಟೆಲಿಗ್ರಾಮ್. ಫಸ್ಟ್ ಇಟ್ ಈಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ನೈಟ್ರೋಜಿನಸ್ ಬೇಸ್. Nitrogenous base. So nucleotide is made up of the first component, which is called the nitrogenous base. Second one is going to be called as the pentose sugar. So ನಿಮಗೆ ಆಲ್ರೆಡಿ ಗೊತ್ತು ಕೆಮಿಸ್ಟ್ರಿಯಲ್ಲಿ pentose ಅಂದರೆ 5 ಕಾರ್ಬನ್ ಅಲ್ವಾ. So it is going to be pentose sugar, means it's a 5 carbon sugar. Okay, it's going to be a 5 carbon sugar. And the last one is going to be called a phosphate group. What is it called? Phosphate group. So students, it's basically PPN. Okay, or NPP also you can remember. NPP, nitrogenous base, pentose sugar and phosphate group together forms your nucleotide. Okay, together forms your nucleotide. ಇವಾಗ ಜಸ್ಟ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಮ್ಯಾಮ್, ಹೌ ಡು ದೀಸ್ ಪೀಪಲ್ ಬಾಂಡ್, ವಾಟ್ ಇಸ್ ದಿಸ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಅಂತ ಡೌಟ್ಸ್ ಇರುತ್ತೆ. ಸೊ ಫಸ್ಟ್ ಹೋಗೋಣ. ಫಸ್ಟ್ ಇಸ್ ಗೋನ್ ಟು ಬಿ ನೈಟ್ರೋಜಿನಸ್ ಬೇಸ್. ಓಕೆ, ಫಸ್ಟ್ ಇಸ್ ಗೋನ್ ಟು ಬಿ ನೈಟ್ರೋಜಿನಸ್ ಬೇಸ್. ನಾ ಸ್ಟೂಡೆಂಟ್ಸ್, ನೈಟ್ರೋಜಿನಸ್ ಬೇಸ್ ಬಂದ್ಬಿಟ್ಟು 2 of them is there. Okay, nitrogenous base is going to be 2. One is called a purine. Okay, I'll write it in different colors so you will be able to differentiate. One is called purine. One is called pyrimidine. Okay, one is called purine, one is called pyrimidine. So these are the 2 nitrogenous base. Okay, these are the 2 nitrogenous base. Okay, now look here. Purine, students, you remember this trick. Pure as gold. Okay, pure as gold. Remember like this, pure as gold. Just remember this trick for now. I'll explain more about it. Next, pyrimidine, you remember cut. CUT. Okay, CUT cut. Now, pure as gold and CUT, we will come into that. But remember that, even though adenu heltare kannadadalli ಹೆಸರು ಚಿಕ್ಕದು ಏನೋ ಹೇಳ್ತಾರೆ ಸ್ಟೂಡೆಂಟ್ಸ್. ದಟ್ ಮೀನ್ಸ್ ನೇಮ್ ಇಸ್ ಸ್ಮಾಲ್ ಬಟ್ ದೇರ್ ಆಕ್ಷನ್ಸ್ ಆರ್ ಬಿಗ್ಗರ್. Something like that. Can anyone tell me and tell me in the chat box what is that? How do I tell it in Kannada? Okay, ಹೆಸರು ಚಿಕ್ಕದು, ಕೀರ್ತಿ ದೊಡ್ಡದು, something like that. Okay, that means name is small but their actions are bigger. So same way here also, purine name is small but it is a 9-membered ring. Okay, it is a 9-membered ring. Okay, 9-membered ring. Got it? 9-membered ring there. Now, look here. On the other side, we have a pyrimidine. Pyrimidine is going to be a 6-membered ring. 6-membered ring. 6-membered ring. Got it? 6-membered ring. Clear? Now, we will learn more about purine and pyrimidine. Okay, we will learn more about purine and pyrimidine. Right now, let me just duplicate this. One second, students. Okay. Ivaga, let's learn first about nitrogenous base. Okay, and then we will come back in learning the other thing. Okay, yes, clear? Oh, what is happening, Preetu? What is this? Quickly smash the like button. Okay, and subscribe to the channel if you are new. So, Nodi, you are going to learn about nitrogenous base. Okay, 1 second. Yeah. Yes, can you all see? Okay, good. So, nitrogenous base, students. Nitrogenous base. So, as I told you, nitrogenous base are going to be, you are going to remember pure as gold. Okay, you are going to remember pure as gold. And what are you going to remember? Cut, right? Pure as gold, matte cut. Okay, so I'll baritini. So, first one is going to be pure as gold. My student's heart is pure as gold. So, atara now we tell no for people. So, students, pure as gold, P stands for, okay, P stands for purine, P stands for purine, A stands for adenine, okay, A stands for adenine, G stands for guanine, okay, G stands for guanine. So, pure as gold, okay, pure as gold, understood? Now, pyrimidine on the other side, what did I tell you to remember? I told you to remember cut, okay, very good. Pyrimidine, I told you to remember the word C-U-T. C for cytosine, U for uracil, T for thymine. Okay, thymine. Ivaga idalli there are some things that you have to learn more. Andre cytosine where it is present, adenine, guanine where it is present. Okay. Now, first term ill nodana students. Cytosine banbitu it is present. Okay, cytosine. 1 second. Cytosine is present both in DNA and RNA. Okay, it is present in both DNA and RNA. On the other side, students, uracil banbitu it's only present in DNA. Thymine is only present in, uracil is only present in RNA, thymine is only present in DNA. Andre, thymine is replaced by uracil. Remember, thymine is replaced, okay, by uracil in RNA. Clear to all of you? Yes or no? Yes, quickly let me know in the chats. Perfect, okay, perfect. Yes, clear? All of you? Give me a thumbs up if all of you understood. And now, as I told you, here, this person, that is your purine, is going to be a 9-membered ring. Okay, 9-membered ring. Okay, on the other side, you have pyrimidine, which is a 6-membered ring. Okay, 6-membered ring. Done? Done? Yes, very nice. Okay. Why can't I see your chats? Okay. Yes, very nice. Okay, Daniel is there. Okay, 6-membered ring. Okay, perfect. So, this is your nitrogenous base for all of you. Now, students, remember, we have more to learn. That is, we have more to learn about their bonding as well, that we will be learning now. Okay, let me try to duplicate this. Yes, got it. Okay, now we will, if we have to learn about your nucleotides. Okay, first we will erase this. Okay, now if we have to think about our nucleotides. Okay, now remember students, how do these nucleotides align? All of that we have to learn, alva. So, this nucleotides you already studied. Okay, nucleotides you already studied that it is going to be NPP. Okay, NPP, that is nitrogen base, pentose sugar and phosphate group. Now, how do they bind? Let's make very simple diagrams. Okay, this is going to be ebox. Okay, this is going to be a nitrogenous base. This triangle banbit nitrogenous base. Okay, color madbidtini. Okay, this is your nitrogenous base. All of you have any doubts in this? 1 second, students. I might put this on myself, this light. Oh, sorry. Okay, done. ಇದು ಹೊಸ ಸ್ಟುಡಿಯೋ, ಸೊ ನನಗೆ ಇಲ್ಲಿ ಯೂಸ್ ಟು ಇಲ್ಲ ಮಾಡೋದಕ್ಕೆ ಕ್ಲಾಸು, ಸೊ ಐ ಆಮ್ ಸ್ಟ್ರಗ್ಲಿಂಗ್ ಅ ಲಿಟಲ್. ಸೊ ನೆಕ್ಸ್ಟ್ ಬಂದುಬಿಟ್ಟು ಪೆಂಟೋ ಶುಗರ್. ಓಕೆ, ಸೊ ಇಲ್ಲಿ ನೋಡಿ, ದಿಸ್ ಇಸ್ ಶುಗರ್. ಓಕೆ, ಐ ಆಮ್ ಜಸ್ಟ್ ಡ್ರಾಯಿಂಗ್ ಬಾಕ್ಸಸ್ ಬಿಕಾಸ್ ಸ್ವಲ್ಪ ಈಸಿ ಆಗುತ್ತೆ ಅರ್ಥ ಮಾಡೋಕ್ಕೆ. ಇವಾಗ ನೀಟ್ ಸ್ಟೂಡೆಂಟ್ಸಿಗೆಲ್ಲ ವಿ ಎಕ್ಸ್ಪ್ಲೇನ್ ವಿತ್ ದ ಹೆಲ್ಪ್ ಆಫ್ ಪ್ರಾಪರ್ ಡಯಾಗ್ರಾಮ್ಸ್. ಓಕೆ, ದಿಸ್ ಇಸ್ ಗೋನ್ ಬಿ ಯೋರ್ ಶುಗರ್ ಆ್ಯಂಡ್ ದೆನ್ ಬೈ ಲಾಸ್ಟ್ ಯು ಹ್ಯಾವ್ ಯೋರ್ ಫಾಸ್ಫೇಟ್. ಓಕೆ, last you have your phosphate. Now let's color that also. Okay, done. Yes, what is this? This is going to be NB, that is nitrogenous base. This is going to be pentose sugar. Okay, sugar, and this is going to be phosphate group. Okay, now students, how do they form the entire dinucleotide? Okay, how do they form? Okay, Sara. Okay, how do they form the entire polynucleotide chain? We have to know. For that, you have to understand the bonding. Okay, you have to understand the bonding. So simple agide students, bonding. First bonding bandu between nitrogenous base and sugar. Okay, between nitrogenous base and sugar, the first bonding is going to happen, which I am drawing it in orange color. So that bond between your nitrogenous base and sugar will be called as glycosidic linkage. What will it be called as? Glycosidic linkage. Okay, glycosidic linkage between a nitrogen base and sugar. Okay, ivaga simple I remember madi, whenever nitrogen base plus sugar, okay, they are together, they will end up forming, students, something called as nucleoside. What will they call? Nucleoside. Side andre when do we tell? When we push someone to the side, alva. So nucleoside andre when we push phosphate. Okay, when we push phosphate out, we call it nucleoside. Okay, we call it nucleoside. Clear to all of you? Yes or no? Okay, now moving forward, NB, that is sugar and nitrogenous base, are going to bind with each other with the help of N-glycosidic linkage. Obviously, phosphate also want to bind, because even though, students, we told nucleoside, when there is NB, okay, plus sugar plus phosphate, okay, plus phosphate, what do we call it? We call it nucleotide, right? Nucleotide. We call it nucleotide. So here it is tied with all the three, and there it is side with just two of them. So whenever side comes, students, suffix banbitu INE anta barutte. Okay, adenosine. Okay, INE anta barutte. Tide alli en barutte 8 anta barutte. Okay, 8. So this, if you want, you can remember, otherwise no need. Okay, yes. Passing package for biology. You still want to pass after exam 3 also? Students, passing in the more marks you should be able to do. Okay, don't just think of passing. We need to get more marks than passing. Okay, classes will come before the biology exam. Nitrogen based sugar. Now, when phosphate has to bind with the sugar, it will bind with the help of, okay, it will bind with the help of phosphoester linkage. What is it called? Phospho. Because phosphate is there, you remember like that only, phosphoester linkage. Okay, phosphoester linkage. Easy na? So far clear all of you? Try to revise. See, first is a nitrogenous base. Nitrogenous base can be purine or pyrimidine. It can be adenine, guanine, cytosine, thymine, uracil, anything. Okay. ಆಮೇಲೆ ಒಂದು ಪೆಂಟೋಸ್ ಶುಗರ್ ಅಂದರೆ ಒಂದು 5 carbon sugar. Okay, it's going to be a 5 carbon sugar. Okay, now this 5 carbon sugar is going to bind with the phosphate on the 5' end. Okay, this is going to be in the 5' end, this is going to be in the 1' end. Okay, 5' to a phosphate with the help of phosphoester linkage. So, ಎರಡ್ bond formation ಇದೆ. One is going to be called the glycosidic linkage, second one is going to be called the phosphoester linkage. Okay. Now, students, whenever a nitrogenous base plus sugar combines together, they are going to form a nucleoside. That means you are pushing someone to the side. Whom are you pushing to the side? You are pushing phosphate to one side. Okay. ಇಲ್ಲಿ ನೋಡಿ, nitrogen base plus sugar plus phosphate together, it is called as nucleotide. Okay, tide. Clear? Yes or no? Okay, which program? The rally. Hey, don't too much ella Uday. It's happening in Bangalore. Wait, news check martini. Yeah, when did this happen? Hey, 37 minutes ago. Oh no. ಆಗಿದೆ ಸ್ಟೂಡೆಂಟ್ಸ್. ಅಯ್ಯೋ. ಓ ನೋ 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 ದಟ್ಸ್ ಬ್ಯಾಡ್ ಯಾರ್. ಇವಾಗ ನ್ಯೂಸ್ ಬಂದಿದೆ ಅಲ್ವಾ, 5:20 ನ್ಯೂಸ್ ರಿಲೀಸ್ ಆಗಿದೆ. ಶೋ. ಓಕೆ, ಯಾ ಕರೆಕ್ಟ್. ಇವಾಗ ಜಸ್ಟ್ ನ್ಯೂಸ್ ನೋಡಿದ್ದೀನಿ. ಓಕೆ, ಬಿಕಾಸ್ ನನ್ನ ಮನೆಯಿಂದ ಬರೋ ಟೈಮಲ್ಲಿ ರ್ಯಾಲಿ, ಪೀಪಲ್ ಆರ್ ಗೋಯಿಂಗ್ ಫಾರ್ ದ ರ್ಯಾಲಿ. ಓಕೆ, ಯಾ, ಸೊ ದಟ್ಸ್ ವಾಟ್, ರ್ಯಾಲೀಸ್ ಆರ್ ವೆರಿ ಸ್ಕ್ಯಾರಿ ಸ್ಟೂಡೆಂಟ್ಸ್. ಸೊ ಇಲ್ಲಿ ನೋಡಿ, NB ಶುಗರ್ ಮತ್ತು ಫಾಸ್ಫೇಟ್ ಗ್ರೂಪ್. ಓಕೆನಾ? ಸೊ NB ಬಂದ್ಬಿಟ್ಟು ದಟ್ಸ್ ವಾಟ್, nitrogenous base binds with the sugar with the help of N-glycosidic linkage. So you can remember like this, sugar is glucose, so glycosidic linkage anta. Okay, now sugar is going to combine with phosphate with the help of what? Phosphoester linkage. So you can remember like this. Okay, phosphate means phosphoester linkage. Okay, now which are the ends at which these people are going to bind? ಇಲ್ಲಿ ನೋಡಿ, ಇವಾಗ ಒಂದು ಸಿಂಪಲ್ ಡಯಾಗ್ರಾಮ್ ಹೆಲ್ಪ್ ಇಂದ ನಾನು ಇದು ಡ್ರಾ ಮಾಡಿದ್ದೀನಿ. ಓಕೆ, ಡ್ರಾ ಮಾಡಿದ್ದೀನಿ. ಸೊ ಇಲ್ಲಿ ನೋಡಿ, ಶುಗರ್ದು ಫಸ್ಟ್ ಕಾರ್ಬನ್ ಇಲ್ವಾ, ಫಸ್ಟ್ ಕಾರ್ಬನ್, ಆ ಫಸ್ಟ್ ಕಾರ್ಬನ್ ಜೊತೆ ನೈಟ್ರೋಜಿನಸ್ ಬೇಸ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬೈಂಡ್, ವಿಚ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ, ವಿ ವಿಲ್ ಯೂಶಲಿ ಟೆಲ್ ಇಟ್ 1,9 or 1,6 ಗ್ಲೈಕೋಸಿಡಿಕ್ ಲಿಂಕೇಜ್. ಓಕೆ, ಆಮೇಲೆ ಈ ಶುಗರ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ಈ ಶುಗರು phosphate group will bind in the 5' end of the sugar. So 1' end, let's imagine this this is my right hand. Okay, program cancelled. Yo, so sad. Did the team come out? Did the team come out of the caravan, like in the bus, or they did not come out? Like before they came only this happened. Uday? So nodi students, nan right hand. Okay, right hand ಬಂದ್ಬಿಟ್ಟು ನಿಮಗೆ ಲೆಫ್ಟ್ ಹ್ಯಾಂಡ್ ತರ ಕಾಣಿಸ್ತಾ ಇರುತ್ತಲ್ವಾ. ಸೊ ರೈಟ್ ಹ್ಯಾಂಡ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ದ ಫಸ್ಟ್ ಕಾರ್ಬನ್. ಓಕೆ, ದಿಸ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬೈಂಡ್ ವಿತ್ whom? This is going to bind with the nitrogenous base. Okay, my right hand is binding with the nitrogenous base. Okay, like this. Now my left hand, that is my 5' end, this is the 5' end, is going to bind with the phosphate. So 1' glycosidic linkage, 5' phosphoester linkage. Clear to all of you? Yes or no? Clear? Okay, very nice. Now let's try and understand nucleoside ಮತ್ತೆ nucleotide ಅರ್ಥ ಆಯ್ತಲ್ಲ. nucleoside ಮತ್ತೆ nucleotide also I hope you have understood. Okay, now let's try to read and understand NCERT. ಅದಕ್ಕೆ ಮುಂಚೆ ನಾನು ಇಲ್ಲಿ ಬಾಂಡಿಂಗ್ ತೋರಿಸ್ತೀನಿ ಸ್ಟೂಡೆಂಟ್ಸ್. ಇಲ್ಲಿ ನೋಡಿ ಬಾಂಡಿಂಗ್. ಓಕೆ, ಇಲ್ಲಿ ನೋಡಿ ಬಾಂಡಿಂಗ್. ಲುಕ್ ಹಿಯರ್ at the bonding, very stood. Okay, okay, oh, okay. So here phosphate in the 5' end, obviously alva, because I told you my 5' end is going to attach to the phosphate. Okay, so phosphate in the 5' end, and then here in the 3'. So students, how is this 5' and 3'? How are we labeling them 5' and 3'? Simple story. Whenever on the 5' end, okay, the 5' end is going to have a free phosphate. Okay, now ivaga illo ulta I have drawn like this like this, you can flip it. Okay, so remember, how are you saying 5' to 3' and 3' to 5'? It's simple. On the 5' end, there is going to be a free phosphate. Okay, on the 5' end, there is going to be a free phosphate. Okay, free phosphate. Clear? On the 3' end, okay, on the 3' end, there is going to be a free OH. From where? From here, from the C carbon. That is, in this E structure really, 5' end ಬಂದ್ಬಿಟ್ಟು phosphate ಜೊತೆ they will join. Okay, so on the 5' end there is obviously going to be a free phosphate, and on the 3' end, so let's consider this is the 5th carbon, this is the 1st carbon. Okay, 1 2. Let's say this is the third carbon. This is here, let's say one OH group is there. This OH, if it's free, we call that end as the 3' end. Okay, 3' end. You don't have much in depth, but for NEET students, we tell a lot. Okay, lot. Okay, now, nodile. This is your NCERT line. Okay, this is your NCERT line. Let us recapitulate the chemical structure of polynucleotide chain. So, students, our polynucleotide 3 components ilva. Adana 1 unit are consider mari. Okay, just consider them as 1 unit. If you consider them as 1 unit, many units like that is going to form your polynucleotide chain. Correct, Alva? It's going to help in the formation of the polynucleotide chain. Correct? Yes or no? Polynucleotide chain. So, like this, 1 nucleotide, 2nd nucleotide, 3rd nucleotide, 4th nucleotide. So, it's entirely going to form what? Polynucleotide. Polynucleotide chain. Like that, many nucleotides. 1, 2, 3, 4, like that, many nucleotides are going to join together. Okay. Now, let's recapitulate the chemical structure of the polynucleotide chain, DNA or RNA. The nucleotide has 3 components. What did I teach you now? The same thing I taught you, that it has 3 components. Okay. A nitrogenous base. So, look here. A nitrogenous base. Okay. A pentose sugar. Okay, that is ribose in the case of RNA and deoxyribose in the case of DNA. Okay, and a phosphate group. Okay, a phosphate group. Clear to all of you? Yes or no? Yes. Okay. There are 2 types of nitrogenous base, purines and pyrimidine. So, purines banbite adenine and guanine. Pyrimidine ಏನು ಬರುತ್ತೆ students? Pyrimidine is going to be cut. Cytosine, uracil ಮತ್ತೆ thymine. Okay, so pyrimidine is going to be cut. CUT. Purine is going to be pure as gold. Okay, now clear agilva idu ಏನದು? Is it better now? Okay, yeah. So, this is clear. Now, cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA at the place of thymine. Idu already explained madidine. Okay. A nitrogenous base is linked to the OH of the first carbon. See, first pentose sugar through an N-glycosidic linkage. So, first pentose sugar jote, okay, first pentose sugar jote, who is going to attach? And nitrogen base is going to attach. Okay, nitrogen base is going to attach, and with the help of what, students? N-glycosidic linkage. N-glycosidic linkage to form a nucleoside. To form what? A nucleoside. Such as, okay, such as. Nan enilva nucleoside andre ine anta barutte end alli. Nodi illi adenosine. Okay, INE bandide. Okay, illu nodi deoxyadenosine. So, nucleoside ige suffix banbiಟ್ಟು prefix banbiಟ್ಟು ine anta. Okay, ine. Clear? INE. So, such as example ಕೊಟ್ಟಿದ್ದಾರೆ adenosine. Okay, deoxy. So, ಅದು RNA ಅಲ್ಲಿ ಬರುತ್ತೆ, DNA ಅಲ್ಲಿ ಬರುತ್ತೆ. ಮತ್ತೆ ಇಲ್ಲಿ ನೋಡಿ guanosine. Okay, deoxyguanosine. Clear? Now, cytidine, deoxycytidine, uridine, deoxythymidine. Okay. Now, when a phosphate group, so second topic ಅಲ್ಲಿ ಹೋಗ್ತಾ ಇದ್ದಾರೆ, when a phosphate group is linked to a OH. So, when a phosphate, so ಇದೆಲ್ಲ I'll just delete. Phosphate group, okay, is linked to the OH of the 5th carbon, okay, 5th carbon of a nucleoside through a phosphoester linkage, okay, through a phosphoester linkage. Clear? Yes or no? Understood? All of you? Yes, ma'am. Okay, very nice. Okay, quickly mark the attendance. I want someone to, take the initiative to make sure that everyone marks the attendance as well, okay? Because your friends are very lazy. They come, they study, they go. Okay, they don't care about all that, teachers and all that. Okay, so I want all of you to ask the students to smash the like button. So, students, look here. N-glycosidic linkage, phosphoester linkage. A corresponding nucleotide or a deoxynucleotide, depending upon the type of the sugar, is formed. 2 nucleotides are linked through 3' to 5', okay, phosphoester linkage to form a dinucleotide. So, what are they telling? Let's say this is a nucleotide and this is a nucleotide. If these two have to bind, it will be bound with a phosphodiester linkage. In students, phosphodiester linkage. Understood? Phosphodiester linkage. Di. 2. Okay. Why? Because there is one nucleotide and another nucleotide. So, idon nucleotide. Ee nucleotide alli 2 bonds ide. Yenalla? N-glycosidic linkage matte phosphoester linkage. Alva? Ee on nucleotide jote join agbekandre, this nucleotide and this nucleotide together they will form the phosphodiester linkage. Okay? Adamele next nucleotide ಆ ತರ ಒಂದು entire polynucleotide chain formation ಆಗುತ್ತೆ. Okay, done. Okay. Now, more nucleotides can be joined in such manner to form your phosphate, phosphate moiety at the 5' end of the sugar, which is referred at the 5' end of the nucleotide. So, ನಾನು ಹೇಳಿಲ್ವಾ 5' end ಅಲ್ಲಿ ಏನಿರುತ್ತೆ? Phosphate ಇರುತ್ತೆ. Okay. At the 5' end, you are going to find a phosphate group. Similarly, on the other end of the polymer, you are going to find a free OH of the 3' group, okay, which is referred to as the 3' end, okay, of the polynucleotide chain. Now, the backbone of the polynucleotide chain is formed due to the sugar and phosphate. Tumba important, students. So, sugar matte phosphate banbitu, it forms the backbone of the DNA. Now, who is going to be between? So, example etkodi, this is sugar, right? My right hand, ee ring irado sugar, okay? Ee ring irado phosphate, okay? They are going to go like this, alva? They are going to wound around like this. Now, imagine this is your nitrogenous base. So, this nitrogenous base is going to project inwards, okay? Project inwards. So, let's say this is your adenine, okay? Adenine will bind with thymine. So, ee tara project inside agutte. This is your sugar, phosphate. They are going to make the backbone, okay? I am telling sugar and phosphate like this, but remember sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, okay? They are going to bind around like this, and these people are going to be facing inwards. Who? The nitrogen bases, okay? Nitrogen bases are going to face inwards. Clear? Now, the nitrogen bases linked to the sugar moiety project from the backbone. Project from the backbone, okay? In RNA, every nucleotide residue has an additional OH group present at the second position. So, students, you have to remember one very important thing, very important thing, okay? Idinna duplicate martini, 1 minute, okay? See, few things I'll add here, okay? I hope we will be able to erase this, students. I'm not sure if we'll be able to. Oh, wait. I don't think we can erase this. Okay, diagrams erase madak chance kammi ide. So we will just delete this. Okay. Now here, let's duplicate. So illi, instead of Chargaff rule, I will just teach you about what we just discussed here. So illi nodi. Okay. So here, what are we going to talk about? We understood that the backbone andre, this is your DNA, okay? This is your DNA structure, okay? In this DNA structure, remember that this bit is making the backbone, and who is going to project inwards? Ma'am, it is so simple that the bases are going to project inwards like this, correct? Bases are going to project inwards. Namage idordu on dod diagram draw madana, students. So let's say I draw a big, okay, magnified image of this. Yellow bandila, on dirty yellow came, okay? Yes. So here, just give me 1 more minute. Yeah. So look here. Let's say I'm drawing this structure, okay, like this. Okay, like this I've drawn the structure, correct? Okay, let me just draw a little more bigger one. See, I'm drawing all of this because I don't want you people to suffer in this chapter, okay? Illandre nanage easy agi chapter complete madbodu, but I want all of you to have that clarity. That's why I'm drawing so much in depth. So, idu backbone, alva? This is the backbone. So, this backbone is obviously, we know what is it made up of. No one has a doubt in that, okay? This is the backbone. Now, students, what is going to project inwards? Let's take an example. E side alli, let's take adenine is there, okay? Then we have cytosine, then we have adenine, then we have thymine. Okay, let's take example like this. Opposite al yaar irtare? Ma'am, opposite banbitu adenine is going to bind with thymine. So T irutte, C is going to bind with G at Gopika Ma'am's class. A is going to bind with T, T is going to bind with A. So this is how they are going to project inwards. Understood? This is how they are going to project inwards. So these are your bases Okay, this is your base pairing. I'm writing BP. Okay. Okay, base pairing. Clear? Base pairing. Done? All of you? Pakka? So, this is the backbone of the DNA and the bases are going to project inwards. Now, remember this very carefully, students. Other than that, what are they telling? They are telling that the RNA, okay, RNA, illi bariyana, okay. Okay, I'm writing it. So, RNA has on the second carbon, okay, on the second carbon, it has an OH group, correct? That is why we call it ribo, right? That is why we call it ribonucleic acid, correct? Ribonucleic acid, yes ma'am. Now, for deoxy, that is for your DNA, what is the situation? On the DNA, okay, on the DNA, the second carbon will have only a H group. That is why we tell, that is why, students, that is why we tell here deoxy. Okay, what do we tell? Deoxy we tell. Here we tell ribo, here we tell deoxyribo. So, because of this, students, we have to remember one thing, that, okay, that we have to remember one thing, that is going to be that this is why this RNA is not stable, because the oxygen will go and bind with everyone. Oxygen will go and bind with everyone, okay? Yes? Clear? Yes? Okay, done. So, this is what that paragraph spoke about. So, I hope this is clear to all of you. Again, I am telling students, don't get confused with nucleoside. Nucleoside is different. This combination is different. Okay, what students write, no? Or tumba easy agi, they write that backbone is made up of nucleoside. Allah. Nucleoside banbitu NB plus sugar. Okay, NB plus sugar andre nitrogenous base plus sugar. Alva. So, that is not going to be the backbone. Sugar and phosphate is going to be the backbone. And as I told you, these people are going to have ine. Okay, ine. Okay, that is why cytidine, adenosine, like example, I will write it here. Example, adenosine. Okay, adenosine. So, like that, adenosine. Understood? Done. So, this is the paragraph talking about. Next one bit, pairing ಬಗ್ಗೆ ಮಾತಾಡಬೇಕು. Okay, I will talk about the pairing. Now, look here. When I talk about purine and pyrimidine, students, I told you pure as gold, okay, and pyrimidine I told you cut, cytosine, uracil, thymine. But you never asked me, ma'am, how these people are going to pair. Is there any rule? Is there any? Yes. So, look here. Remember, students, let's take a classroom, okay, idon classroom, okay, idi innond classroom. So, this is, let's say, A, 10A and 10B, okay, 10A and 10B. This classroom ali, let's say there are, okay, 5 boys. There are 4 boys. 4 boys are there, okay. Now, I want to do a dance, okay, I am organizing a dance for the annual day, and I notice that this class has like 4 boys, and this class, okay, this class has like 3 girls, okay, 3 girls, and this class has this 1 girl. Okay, this class has this 1 girl. So, ನಾನು ಏನು ಮಾಡ್ತೀನಿ ಮ್ಯಾಮ್, ನಾರ್ಮಲಿ ನೀವು ಏನು ಮಾಡ್ತೀರಾ ಮ್ಯಾಮ್, ಆ 4 ಬಾಯ್ಸು, ಓಕೆ, you will take all the 4 boys, okay, and you will take all the 4 girls from both the sessions, right? So, this is how I can do a dance. I can do a dance only if I have proper pairing. So, these are going to be a pair, these are going to be a pair, these are going to be a pair, and these are going to be a pair, and automatically my dance will happen. So, students, it is understood that how many boys are there, that many girls also should be there for my dance to happen. Let's take example, illi, let's say 5 girls are there and here only 4 boys are there. Pairing tumba kashta agutalva, 1 girl will be left alone. So, same thing happens here also. Remember that whenever we talk about purine and pyrimidine also, whenever we talk about who? Purine and pyrimidine. Okay, purine and pyrimidine, we should remember that how much purine is there, that much pyrimidine should be there, otherwise bonding is not going to happen. Okay, otherwise bonding is not going to happen. So, ill example etkolna. So, let's say adenine will bind with who? Adenine will always bind with thymine. With who? Adenine will always bind with thymine with the help of 2 hydrogen bonds. With the help of what, students? 2 hydrogen bonds. Okay, tumma important. 2 hydrogen bonds. 2 hydrogen bonds. Done? Clear to all of you? Yes or no? 2 hydrogen bonds. Done? Okay. Now, illi next example banbitu. Guanine will bind with cytosine. Okay, guanine will bind with cytosine with 3 hydrogen bonds. With 3 hydrogen bonds. Okay, so this is rule. Okay, you cannot change this rule. Okay, you cannot change this rule. So, students, to remember this, I'll give you a trick. Remember, at Gopika Ma'am's class. Okay, remember like this, at Gopika Ma'am's class. So, you'll never go wrong. AT at alva. So, adenine always bind with thymine. Guanine will always bind with cytosine. So, remember like that. Okay, at Gopika. Guanine is going to bind with cytosine with 3 hydrogen bonds. So, yavat difficult agutte break madake, the guanine and cytosine will be a little difficult to break. Okay. So, this is what Chargaff told, but Chargaff rule is more than that, students. Chargaff told that the ratio of purine should be equal to the ratio of pyrimidine. So, that is what I told you here. So, according to Chargaff, okay, according to Chargaff, that is why I have written here Chargaff rule, ratio of purine should always be equal to the ratio of pyrimidine. Okay, ratio of purine should always be equal to ratio of pyrimidine. Allandre binding properly agalla alla. ಎಷ್ಟು adenine ಇದೆ ಅಷ್ಟು guanine ಇರಬೇಕು, ಅಷ್ಟು ಎಷ್ಟು adenine ಇದೆ ಅಷ್ಟು thymine ಇರಬೇಕು, ಎಷ್ಟು guanine ಇದೆ ಅಷ್ಟು cytosine ಇರಲೇಬೇಕು. Okay, yes, okay, clear to all of you? Yes or no? Done? Pakka? Okay, now we have to learn more about I'll make you solve one problem also here. Okay, I'll make you solve one problem also here. 1 minute. Okay, students, run class alli NCERT full. I will copy paste NCERT for you people, so don't worry about NCERT. This is specially for my PUC students. Okay, PUC CBSE class 12 students. Okay, ill nodi students. So simple. Okay. Simple andre they will give you problems for exam. They will give you that, let's say if adenine, okay, adenine is 30%, okay, find cytosine. Ee tara question kotre heng martira. Okay, think ಮಾಡಿ ಎಲ್ಲರೂ. Think and tell me if such a question comes. Okay, if such a question comes, tell me how will you find it. ಬೇಗ ಚಾಟ್ ಬಾಕ್ಸ್ ಅಲ್ಲಿ ಸ್ವಲ್ಪ ತಿಳಿಸಿ. ಓಕೆ, yes, come on. Done? Try and calculate. Try and calculate faster. Okay. Okay. Now, look here. So, if adenine is 30% and cytosine, you have to find cytosine. ನಿಮಗೆ ಏನು ಗೊತ್ತು, adenine thymine ಜೊತೆ ಬೈಂಡ್ ಆಗುತ್ತೆ ಮ್ಯಾಮ್, adenine ಮತ್ತೆ thymine ವ್ಯಾಲ್ಯೂ ಬಂದ್ಬಿಟ್ಟು ಸೇಮ್ ಆಗಿರಬೇಕಂತ ಅಲ್ವಾ. So remember like this, students, if adenine, okay, if A is equal to 30%, then it is understood that even T should be 30%, correct? Even T should be 30%. Otherwise, how will I make A to T, right? So this is correct. Now, here almost 60% is over. Now, what they have asked you to find cytosine. So, let's say cytosine will be 60. So, 60 minus will be 40. So, 20% will be cytosine. If cytosine is 20%, obviously guanine also has to be 20%. So, this is how you are going to do the maths. Yen question keldreno, if rule, you have to remember the rule. Chargaff rule. Adenine 30% kotre, thymine will be 30%. Cytosine 20% idre, guanine will be 20%. Okay? Clear? Yes, very good, Rohit. Very good. So, cytosine is going to be 20%. Okay, 20%. So, that is how your numericals are going to be. Clear to all of you? Yes or no? Okay. 15 people are. Seriously? Yes, very good, Rohit. Oh, no. So bad, so bad. Okay, come on. Now, the salient features of double helical structure of DNA is as follows. No, so, salient features banbit tumba important, okay? Tumba important. So, remember, it is made of 2 polynucleotide chain, whereas the backbone is constituted. So, students, DNA banbit eno 2 polynucleotide chain, alva? 2 polynucleotide chain. So, one chain, another chain. Okay, one chain and another chain. So, 2 polynucleotide chain. These 2 polynucleotide chains where the backbone is constituted by a sugar and phosphate group. This also I taught you now. And bases are projecting inside. So, this is the first point which you already studied now. Okay. The 2 chains have anti-parallel polarity. Anti-parallel polarity andre eno? I'll show you one second. So, let's say illinda illon chain, illinda illon chain. Each chain banbit 3' to 5' idre, ಇನ್ನೊಂದು chain banbit 5' to 3'. Okay, so anti-parallel polarity. Okay, sorry, anti-parallel polarity. Anti-parallel polarity. It means one chain has 5' to 3' means the other has 3' to 5'. The bases in 2 strands. The bases in 2 strands are paired through the hydrogen bonds. This also you know. Just now you studied. If adenine is there, adenine will bind with thymine with 2 hydrogen bonds. Okay, in the between 2 hydrogen bonds is there. If guanine is there, guanine will bind with cytosine with 3 hydrogen bonds. So, obviously, in between 3 hydrogen bonds are there. Okay, I am drawing it this way just to show you the explanation. The bases in the 2 strands are paired through hydrogen bonds forming base pairs. So, a hydrogen bond, if it is there in the middle only, we can call them as base pairs. Okay. Now, adenine forms 2 hydrogen bonds with thymine from the opposition strands and vice versa. Generally, guanine is bonded with cytosine with 3 hydrogen bonds. Okay, 3 hydrogen bonds, and here we have 2 hydrogen bonds. Now, as a result, always a purine comes opposite to a pyrimidine. This generates approximately uniform distance between 2 strands of the helix. Now, imagine, students, it is normal, alva? It's common sense to understand that ivaga adenine, thymine, guanine, cytosine bandre, okay, adu standard agirutte. Tumba changes bandre, middle alli space jaasti agutte, alva? So, both the helix will not be, like spacing will not be correct, okay? Now, the 2 chains are coiled, okay? The 2 chains are coiled in a right-handed fashion, okay? In a right-handed fashion, The pitch of the helix. Now, what is the pitch of the helix? Pitch meaning andre, pitch of the helix andre, 1 complete turn, okay? 1 complete turn. 1 complete turn, okay? The pitch of the helix is 3.4 nanometers. So, 1 complete turn andre, 3.4 nanometer. Okay, 3.4 nanometer. A nanometer is 1 billionth of the meter. So, that is going to be 10 to the power minus 9. Namaga convert madbekandre 3.4 into 10 to the power minus 9 meter anta baribodu. Okay. Okay, and there are roughly 10 base pairs. So, students, ee one ternally, like let's imagine itara on DNA. Okay. So, one ternally, okay, one whole thing will have 10 base pairs, okay, 10 base pairs. So, that is also important for you to remember, 10 base pairs in each of the turns, okay, in each of the turns. Now, salient feature, so, students, this is the pitch, 1 complete turn. So, in that 1 complete turn, you are going to have 10 base pairs, okay, in 1 complete turn, you are going to have 10 base pairs. So what and all are you going to write for the salient features of DNA if they ask you question? You are going to start with the backbone. Okay, you start with the backbone. Okay, I'm trying to write everything for you people. So first you start with the backbone. Who makes the backbone? Sugar plus phosphate makes the backbone, and the bases, okay, bases project outwards. Okay, projects. Actually, it's inwards. Projects inward. Okay, projects. Now, next is going to be what? That is the first point. Second point, you are going to write about the base pairing, or you can write about the anti-parallel polarity. Okay, anti-parallel polarity. Andre, one is going to be from 3 dash to 5 dash. Okay, another one is going to be 5 dash to 3 dash. Okay, this is the anti-parallel polarity. Third point, ಏನು ಬರಿಬೋದು students? ನಮ್ಮ bonding ಬರಿಬೋದು. Adenine will bind with thymine with 2 hydrogen bonds. Guanine will bind with cytosine with 3 hydrogen bonds. And this is important. This is the base pairing. Okay. I'm only writing the keywords so you can remember and write it in the exam. Okay. Now, fourth point, students, you have to write about the pitch. Okay. Pitch of helix is going to be 3.4 nanometer. Okay, 3.4 nanometer, and in one turn. Okay, in one turn. Hi, silent heart. Okay, in one turn. Okay. Aishu? Is it Aishu? Okay, in one turn we are going to have 10 base pair. Okay, 10 base pair. Understood? One turn, 10 base pair, and 3.4 nanometer. Okay, 3.4 nanometer. So you can write 3.4 into 10 to the power minus 9 meter also, because nanometer to meter you have to convert it. Okay, if you want for any calculation. Done? Done. Okay, so this is your salient features of DNA. Okay, salient features of DNA. You'll notice they have also told that consequently the distance between a base pair in a helix is approximately 0.34 nanometer. Simple, students. 3.4 divided by 10 madbidi. 3.4 ಏನು pitch of the helix ಅಂದ್ರೆ one turn. Okay, one turn ಇಲ್ಲಂದ್ರೆ pitch of the helix. Okay, pitch ಅಂತ ಬರೀತೀನಿ. Okay, pitch divided by the number of base pairs. So that is going to be 0.3. Okay, 0.3. Ma'am, you blocked me. When did I block you? Chumma, don't tell nonsense. Okay, I did not block anyone. I am standing here and teaching. Okay, students, 10-15 people are sitting on a channel. Okay, anyone can monitor the chats. It's not me who monitors the chat alone. Clear? It's not my channel. It's many people's channel. Okay, all our team members, all our back-end team, everyone are checking the chats. Okay, so why will I do it? What happened? Who removed you? You're able to message no. Not coming. What happened? Did you like talk in any other class? Like, I don't know, students. I can see your messages. Okay, if like some other person has removed you, then I don't know what is the thing. Okay, okay, I have not done it. I can see your messages. Sometimes it happens, students. What happens? No, many people are watching. Like many people are checking. No, sometimes like that by mistake. Like maybe before you there were a spammer. So by mistake, instead of clicking that person's name, and next person's name can get removed or like muted or timed out. So that is very normal. If you have another account, you can come from that. I will go and check. In case your name is there, I'll get it removed. Okay. Okay. Now the plane of one base. Okay, the plane of one base pair stacks over the other in a double helical. This in addition to the H bond confers stability. Okay, confers stability of the helical structure. And one above the other, one above the other, one above the other, one above the other. This makes the entire DNA helix stable. Okay, entire the DNA helix stable. Clear? So you'll notice, students, whatever we studied so far, this diagram is there. Okay, whatever we study so far, it is diagram, it's there. Okay, so here you see what is there. This is one entire nucleotide. Okay, this is one entire nucleotide. Correct? Entire nucleotide. Phosphate is there, your sugar is there, and your entire bonding is there. And here you have your nitrogen bases. Okay, this is your nitrogen bases. Bonding is going to happen with 2 hydrogen bonds. Okay, 2 hydrogen bonds. And here you have thymine, again a nucleotide. So, ee tara erad chains madhyadalli. Okay, erad chains madhyadalli you will be able to see the base pairing. Okay, BP I am writing, base pairing you will be able to see, and here you will be able to see your backbone. Okay, backbone andre sugar plus phosphate. Clear? All of you understood? All of you understood? Done? Okay, yes, understood. Clear? This is your backbone. This is the backbone. In between you have your nitrogenous bases in between. Okay, nitrogenous bases in between. Done? Okay, break beka tumba aita. Okay, central dogma I'll come and do. Okay, one kutti break we can take. Okay, because I think all of you are very much disturbed with the news. Okay, what is the time, students? 6:19 aitala. Okay. Okay, after this no break. Okay.
सेशन तो नहीं है ना, लिखा था सेशन 12।
Okay. Continue madana students. Hello. Let's continue. Okay, ready? So we have done up to, understanding the structure. Okay. Ivaga we have to learn about central dogma. Okay, central dogma ಬಗ್ಗೆ we have to study. Okay, let's just erase this. Okay, now what is central dogma? I'll tell you what is central dogma. So listen to me carefully, all of you. Okay, listen to me carefully, all of you. Central dogma means this is everything of our molecular biology. ಇವಾಗ ನಾನು ಏನು ಬಗ್ಗೆ ಮಾತಾಡ್ದೇನು ಮಾಲಿಕ್ಯುಲರ್ ಬೇಸಿಸ್ ಆಫ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಅಲ್ಲಿ ಎವ್ರಿಥಿಂಗ್ಸ್ ದ ಕೋರ್ ಫೌಂಡೇಶನ್ ಇಸ್ ಸೆಂಟ್ರಲ್ ಡಾಗ್ಮಾ. ದಟ್ ಈಸ್ I need to ultimately make my protein. Proteins could be different types of protein. It could be regulatory, it could be, helping in something, it could be the one that has some, wholesome characters. So, ultimately, ನನಗೆ protein synthesis ಮಾಡಬೇಕಂದ್ರೆ I have to go through this entire central dogma. So, let's understand what is the central dogma. Okay, DNA, students, will become RNA. RNA will become protein. RNA will become protein. Correct, Alva? Now, DNA to RNA. Okay, DNA to RNA. Munche namu gottirbeku. DNA can form its own copies. Okay, DNA can form its own copies with the help of a process called as replication. Replication. Okay, replication. DNA can form its own replica with the help of a process called as replication. Okay. Next, DNA to RNA. DNA to RNA. This process ನಾವು ಏನು ಕರೀತೀವಿ students? We call it transcription. Transcription. Clear? DNA to RNA, we call it what? Transcription. RNA to protein ಗೆ translation, because ಇಲ್ಲಿ no translate ಆಗ್ತಾ ಇದೆ. Okay, translation. So, this whole thing, students, is called as the central dogma. We call it what? The central dogma. Clear? We call this the central dogma. This full process, this entire chapter is there. DNA to RNA is called transcription. RNA to protein is called translation. DNA can form its own copy with the help of transcription. DNA can form its own copy with the help of transcription. Okay, transcription. So, DNA can form its own copy with the help of replication. Okay, this is your entire central dogma. Entire central dogma. Okay, clear to all of you? Yes or no? Now, let's try and understand one more very important concept called packaging of DNA. And let me tell you, if you study this concept right now, it's going to be so easy, students. This entire chapter is going to be easy. So, students, simple question ಕೇಳ್ತೀನಿ. ಒಂದು ದೊಡ್ಡ ಥ್ರೆಡ್, okay, ಒಂದು ದೊಡ್ಡ ಥ್ರೆಡ್, approximately 2.2 meter long thread. ಇವಾಗ I am asking you to put it into a small nucleus. Will it be possible? Will it be possible for you to put this entire thread into this small nucleus? You will be like, ma'am, what's wrong with you? Obviously, it is not possible. Alva, you will tell me, ma'am, obviously, it's not possible. So, I'll be like, okay, let's try some other method. Right, let's try some other method. So, students, ಇದೇ ಸೇಮ್ ರೀಸನಿಂದ ಈ ಡಿ ಎನ್ ಎ ಬಂದ್ಬಿಟ್ಟು 2.2 meter ಲಾಂಗ್ ಒಂದು ಡಿ ಎನ್ ಎ ಅಪ್ರಾಕ್ಸಿಮೇಟ್ಲಿ. ಆ ಡಿ ಎನ್ ಎ ನಾವು ನ್ಯೂಕ್ಲಿಯಸ್ ಒಳಗಡೆ, ಓಕೆ, ಇಫ್ ಯು ಹ್ಯಾವ್ ಟು ಪ್ಲೇಸ್ ಇಟ್ ಇನ್ಸೈಡ್ ದ ನ್ಯೂಕ್ಲಿಯಸ್, ಆಬ್ವಿಯಸ್ಲಿ ಇಟ್ಸ್ ನಾಟ್ ಪಾಸಿಬಲ್ ಚುಮ್ಮಾ ಇಫ್ ಐ ಲೈಕ್ ರೋಲ್ ಇಟ್ ಆ್ಯಂಡ್ ಕೀಪ್ ಇಟ್ ಇನ್ಸೈಡ್ ಅಲ್ವಾ. ಅದಕ್ಕೊಂದು ಸಪ್ರೇಟ್ ಒಂದು ಪ್ಯಾಕೇಜಿಂಗ್ ಟೆಕ್ನಾಲಜಿ ಇದೆ, ಒಂದು ಸಪ್ರೇಟ್ ಒಂದು ಪ್ಯಾಕಿಂಗ್ ಮೆಕ್ಯಾನಿಸಮ್ ಇದೆ. ಸೊ ಆ ಪ್ಯಾಕಿಂಗ್ ಮೆಕ್ಯಾನಿಸಮ್ ಏನಂದರೆ, ಡಿ ಎನ್ ಎ ಬಂದ್ಬಿಟ್ಟು ನೆಗೆಟಿವ್ಲಿ ಚಾರ್ಜ್ಡು. ಡಿ ಎನ್ ಎನು ನೆಗೆಟಿವ್ಲಿ ಚಾರ್ಜು. ಈ ನೆಗೆಟಿವ್ಲಿ ಚಾರ್ಜ್ ಡಿ ಎನ್ ಎನ ನನಗೆ ಅದನ್ನು ಪ್ಯಾಕ್ ಮಾಡಬೇಕಂದ್ರೆ ನಾನು ಆಬ್ವಿಯಸ್ಲಿ ಪಾಸಿಟಿವ್ ಚಾರ್ಜ್ ಏನಾದರೂ ಮೆಟೀರಿಯಲ್ ಯೂಸ್ ಮಾಡ್ತೀನಿ ಅಲ್ವಾ. ಸೊ ಐ ಆಮ್ ಗೋಯಿಂಗ್ ಟು ಯೂಸ್ ಅ ಪಾಸಿಟಿವ್ಲಿ ಚಾರ್ಜ್ಡ್, ಓಕೆ, ಐ ಆಮ್ ಗೋಯಿಂಗ್ ಟು ಯೂಸ್ ಅ ಪಾಸಿಟಿವ್ಲಿ ಚಾರ್ಜ್ protein, okay, a positively charged protein which is called histone proteins. What is it called? Histone protein. Clear? Histone proteins. Okay, So to pack this negatively charged DNA, I am going to take the help of positively charged protein called as the histone proteins. Ma'am, how does this entire thing happen? Let me explain. So students, only one histone proteins will come for packaging? Illa. Okay, almost 5 histone proteins ಬರ್ತಾರೆ. Okay, H2A, H2B, H3, H4 ಮತ್ತು H1 ಪ್ರೋಟೀನ್. ಇಷ್ಟು ಪ್ರೋಟೀನ್ ಸೇರಿಬಿಟ್ಟು ಈ ಡಿ ಎನ್ ಎ ಪ್ಯಾಕ್ ಮಾಡೋಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತಾರೆ. ಹೆಂಗೆ ಮ್ಯಾಮ್? ತೋರಿಸ್ತೀನಿ ಸ್ಟೂಡೆಂಟ್ಸ್. ಸೊ ನೋಡಿ, ನಮಗೆ ಒಂದೊಂದು ಕಲರ್ ಎತ್ಕೊಳ್ಳೋಣ. ಓಕೆ, ದಿಸ್ ಈಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ಯೋರ್ H2A. ಓಕೆ, ಇದನ್ನು ಕಲರು ಮಾಡಿಬಿಡೋಣ. ಓಕೆ ಓಕೆ, ದಿಸ್ ಈಸ್ ಯೋರ್ H2A. ಓಕೆ, ನೆಕ್ಸ್ಟ್ ಈಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ H2B. ಓಕೆ ಓಕೆ, this is your H2B. okay, ಇದನ್ನು ಕಲರ್ ಮಾಡೋಣ. correct? okay, H2B. now next is going to be H3. okay, H3. so if I draw it with colors, you will understand faster. That's why I'm using colors. okay, H3. okay, last one is going to be H4. Don't ask me about H1, I will tell you about H1. okay, H1 is suspense thriller. okay, done. So these are the 4 proteins. okay, H2, so I'll write it here. okay, H2A, okay, H2B, H3 ಮತ್ತೆ H4. okay, ಈ 4 proteins ಇದಲ್ವಾ? ಬಟ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ಈ 4 proteins ಇಂದ ಆಗಲ್ಲ. what we are going to do? We are going to take each of them 2. One more. okay, one more. okay, one more and one more. so ಇವಾಗ ಎಷ್ಟಾಯಿತು ಸ್ಟೂಡೆಂಟ್ಸ್? ಟೋಟಲ್ ಹೇಳಿ ಎಷ್ಟಾಯ್ತು ಅಂತ ಬೇಗ tell me how much is the total quickly in the chats. total in the chats. okay, 1 minute. okay, so okay, done. now you are left with orange and blue. You'll be thinking, ma'am, why are you sitting in coloring? Coloring helps students. My students remember the colors at which I drew. okay, they don't remember what I wrote. More than that, they remember the color. so I think you people also will be able to work accordingly. okay, so you look here. how many are there? Come on, quickly, let me know. Who is that AJ who cannot see if it is ma'am or sir? okay, sir. sir, it seems he's in some other world. okay, so this total makes how many students? 1 2 3 4 5 6 7 8. ma'am, total ಬಂದ್ಬಿಟ್ಟು 8 histones together. so 8 andre octa anta kariboda students? so I am going to call this entire thing as histone octamer. what am I going to call it as? histone octamer. okay, histone octamer. so this histone octamers are going to help me. okay, this histone octamers are going to help me, very good Shreya, to make the complete DNA packaging. how ma'am? I will tell you that also. so students, the entire histone octamer na namak pink on dot consider madana. okay, full octamer na on dot consider madana. so hengidu full binding will happen. idon nim long DNA alva. ee long DNA madhyadalli like this one entire histone octamer will be there. okay, like this one entire histone octamer will be there. again you can see the winding going like this. okay, again one histone octamer itara continuous like this to pack the DNA strands. okay, see, I am explaining to you, that's why I am drawing it like this. illandre proper condensation nodbodu. okay, proper condensation nodbodu. so again like this, again we will have, okay, like this, many will be there. okay, like this, and then they will condense like this, students. They will condense like this. So in that also obviously your histones are going to be there. Okay, one second. Okay, like this histones, around the histones it is going to wound. Okay, and ultimately they are going to help in the formation of your chromatin fibers. Okay, they are going to help in the formation of the chromatin fibers. Itara, ultimately what will they form? Your chromosomes. Okay, itara, it will form the chromosomes. Clear? Clear? Condensation is clear to all of you? Yes or no? Yes or no? So here what is going to happen? This entire DNA strand is going to wound around histones, because the entire histone octamer na on ping ball consider ಮಾಡಿದೀವಿ. So a ping ball like what it is, rolling that, wounding that, we have a DNA strand, and again after condensation they are going to ultimately form the chromosomes. Okay. Now students, histone octamer, okay, remember this, histone octamer plus DNA ilva, idina together we will call it as nucleosome. What will we call it as? Nucleosome. Okay, that is, we will have histone plus DNA together is called the nucleosome. Clear to all of you? Yes or no? Yes, share madi vega vega. Okay, Aishu is telling, so share all of you. Aishu is like the class leader. Okay, taken the distance between two consecutive base pair as 0.3 nanometer. Okay, how did we get 0.3 nanometer? Do you all understand? That time only I told you, pitch of the helix is 3.4 divided by the number of base pairs between them, which is going to be 0.34. Okay, so that is how we got the distance. Okay, so if the length of the DNA double helical is typically mammalian cell is calculated by multiplying the total number of base pairs with distance between two consecutive base pairs, it is going to be 6.6 into 10 to the power 9 into 0.3 into 10 to the power minus 9. So apparently ಎಷ್ಟು ಬರುತ್ತೆ students? Simple ಆಗಿ ಮಾಡಿ 6.6 into 10 to the power 9 base pair into 0.34 into 10 to the power minus 9. Okay, ಇದು ಕಟ್ ಮಾಡೋಣ. ಅದಾದಮೇಲೆ ಇದು ಮಲ್ಟಿಪ್ಲೈ ಮಾಡಿದ್ರೆ ಎಷ್ಟು ಸಿಕ್ಕುತ್ತೆ? 2.2 approximate. Okay, 2.18 ಅನ್ನೋ ಸಿಕ್ಕುತ್ತೆ. So 2.2 approximate ಅಲ್ವಾ? So quickly mark your attendance. Come on, all of you. Very nice energy. Okay. So a length that is far greater than the dimension of a typical nucleus. So typical nucleus alli approximately 10 to the power minus 6 okay meter irutte approximate size. ಆ ನ್ಯೂಕ್ಲಿಯಸ್ ಒಳಗಡೆ 2.2 ಮೀಟರ್ ಲಾಂಗ್ ಒಂದು DNA ಸ್ಟ್ರಾಂಡ್ ಹಾಕೋದು ಇಂಪಾಸಿಬಲ್. ಸೊ ಅದಕ್ಕೆ ನಾವು ಪ್ಯಾಕೇಜಿಂಗ್ ಅಂತ we are doing. So in prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. Imagine ಮಾಡಿ students, ಒಂದು ದೊಡ್ಡ ಥ್ರೆಡ್ ಕೊಡ್ತಾ ಇದ್ದೀನಿ ಇಲ್ಲಿ ಅಂದರೆ ಒಂದು ದೊಡ್ಡ ರೋಪ್ ಕೊಟ್ಬಿಟ್ಟು ಒಂದು ಚಿಕ್ಕ ರೂಮಲ್ಲಿ ನಿಮ್ಮನ್ನ ಲಾಕ್ ಮಾಡಿದ್ರೆ ಏನಾಗುತ್ತೆ, ಆ ರೋಪ್ ಎಲ್ಲ ಕಡೆನೂ ಸ್ಪ್ರೆಡ್ ಆಗುತ್ತಲ್ವಾ. So they are telling, even though prokaryotes do not have a proper nuclear membrane, the DNA is not scattered. Okay, so DNA is held with some proteins in region called as nucleoid. Okay, the DNA in the nucleoid is organized in the large loops held by proteins. Which are the proteins? Histone proteins. Okay, now in eukaryotes this organization is much more complex. There are a set of positively charged basic proteins called as histones. So nimigella gottu, proteins are made up of amino acids. Okay, proteins are made up of amino acids alva, and here they are talking about two basic proteins which amino acids we will come into. The protein acquires charge depending on the abundance of amino acids. Okay, amino acids ಜಾಸ್ತಿ ಇದ್ರೆ obviously these people are going to be positively charged. So depending on the amino acids. Okay, residue with charged side chains. Histones are rich in basic amino acid residue. So histones are rich in basic amino acid residue lysine and arginine. So ಯಾವ್ದಲ್ಲಿ ರಿಚ್ ಆಗಿರುತ್ತೆ? ಲೈಸಿನ್ ಮತ್ತೆ arginine. Okay, so it is rich in lysine, MCQ question can come, and arginine. Lysine and arginine. Okay, both the amino acid residue carry positive charge in their side chains. Histones are organized to form a 8 molecule. See, unit of 8 molecules called the histone octamer. Did all of you understand? Every line of NCERT I am teaching all of you. Okay, now the negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called as nucleosome. Okay, a typical nucleosome, students, contains 200 base pairs of the DNA helix. So, on nucleosome ಎಷ್ಟು base pairs ಇರುತ್ತೆ? Base pairs ಏನು? Adenine, thymine, so guanine, cytosine. So, 200 base pairs ಇರುತ್ತೆ. Okay. Nucleosome constitute the repeating unit of structure in the nucleus called as the chromatin. So, ಆ nucleosome continuous ಆಗಿ condense ಆಗಿ chromatin form ಆಗುತ್ತೆ. Ultimately, ನಿಮ್ಮ chromosome. ಇವಾಗ ನೀವು 12th ಅಲ್ಲಿ when you go to the lab, you will be doing prophase, metaphase, anaphase, telophase. ಅದರಲ್ಲಿ ನೀವು prophase ನೋಡಿದ್ರೆ student, generally ನಿಮ್ಮ prophase ಬಂದ್ಬಿಟ್ಟು ಈ ಥರ ಇರುತ್ತೆ. Okay, it is going to look something like this, okay, and inside you will notice like this. Wait, I will make it very thin and show you people. Okay, it will look like this, something like this. Okay, have anyone seen it? Like this your prophase is going to look like. Okay, slowly in metaphase you will start seeing like this chromosomes. Okay, like this. So what happened? Because condensation happened. Okay, so you'll notice the nucleosomes in the chromatin are seen as a bead on a string structure. Okay, it is seen as a bead on a string structure. Beads on a string structure. Andre example il nodi students. So microscopically, let's say ide nan view. Okay, microscopically this is my field. Okay, in that field how will it look like? It will look something like this. Okay, okay, like this. Now like this andre in between there are going to be, okay, wait, let me just draw another color. In between there are going to be dots like this. Okay, so it looks like a beads on a string structure. Beads ilva, malayed beads atara. Okay, beads on a string structure. Clear? Done. Now look here, when viewed under the electron microscope, so microscope al nodidre ee tara irutte. Okay, done. Yes. Okay, now theoretically how many such beads do you imagine are present in the mammalian cells? The beads on a string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at the metaphase. Adike students, prophase alli ee tara irutte nim diagram. Metaphase alli suddenly nimage slowly chromosomes you will start seeing. Why? Because further coiling will happen in the metaphase. Okay, further coiling will happen in the metaphase. Oh, thank you Satvik. Okay, moving forward. Non-histone. So, ಇಲ್ಲಿ ಸ್ವಲ್ಪ ಇಂಪಾರ್ಟೆಂಟ್ ಟಾಪಿಕ್ಸ್ ಇದೆ ಸ್ಟೂಡೆಂಟ್. ಸಿಂಪಲ್. ಇವಾಗ ನನಗೆ ಒಂದು ಅಡಿಷನಲ್ ಹೆಲ್ಪ್ ಬೇಕು. ಲೈಕ್ ಮ್ಯಾಮ್, I cannot teach. Okay, I'll say that I cannot teach this chapter. I need Baswa sir also to come here, or I need one more additional person to come and help me. When I'm saying that, it is understood that I need some more people to help me. So here also students, packaging ಮಾಡಬೇಕಂದ್ರೆ ನನಗೆ ಒಂದು ಅಡಿಷನಲ್ ಹೆಲ್ಪಿಗೆ ನಾನೊಂದು ಅಡಿಷನಲ್ ಸೆಟ್ ಆಫ್ ಪ್ರೋಟೀನ್ಸ್ ನ I'll be utilizing, which is called as your non-histone chromosomal protein, which is called as your NHC. What is it called? NHC. These are the additional set of proteins that I'm going to take help for. Okay, in the case of your euchromatin. Okay. Okay, additional set of proteins. Okay, additional set of proteins that I will take to, you know, for the entire binding to happen, which is NHC, non-histone chromosomal protein. Okay, understood? Ivaga illi two things they are talking about, euchromatin matte heterochromatin. Okay, so just take a chromosome like this. Okay, like this. Okay, this is one chromatid. Okay, this is one. Ivaga E on chromosome. See, the chromatin that is more densely packed and stained are called euchromatin. In a typical nucleus, some region of the chromatin. Okay, so let's assume students, see, some region of the chromatin will be tightly packed and some region will be loosely packed. Again, some region will be loosely packed, some region will be tightly packed. Alva, correct? Okay, ill nodi. So obviously students, namak already artha agutte that tightly packed region alva, tightly packed region ge ನಾವು heterochromatin, we call it what? Heterochromatin. So obviously it's understood, if a room is tightly packed with people, let's say ಈ ಒಂದು ಚಿಕ್ಕ room ಅಲ್ಲಿ 50 people are coming and sitting, ಅಲ್ಲಿ ನಾನೊಂದು holy color, like let's say I am throwing one color, ಎಲ್ಲರ ಬಾಡಿಯಲ್ಲೂ ಸ್ಪ್ರೆಡ್ ಆಗುತ್ತೆ ಅಲ್ವಾ, ಬಿಕಾಸ್ ಎಲ್ಲರೂ ಕಂಜೆಸ್ಟೆಡ್ ಆಗಿದೆ. ಸೊ obviously when I am throwing something, it will fall on everyone. So heterochromatin students, here they are going to be densely stained. densely stained. Clear? Densely stained. Clear? Yes or no? Okay. And they are transcriptionally inactive. Okay. They are transcriptionally inactive. Okay, transcriptionally inactive. Now, adhe chromosomally, students, adhe chromosomally, there will be regions where they are loosely packed. Alva, there will be places where they are loosely packed. So, il nodi, idu loosely packed. Loosely packed. Ee loosely packed region ge naam ಏನು ಕರಿತೀವಿ? Euchromatin ಅಂತ ಕರಿತೀವಿ. Euchromatin. Okay. Euchromatin ಬಂದ್ಬಿಟ್ಟು students lightly stained. Okay. Lightly. ಅದು dense ಇದ್ರೆ obviously ಇದು light ಆಗೇ ಇರಬೇಕು. So lightly stained. ಅದೇ example ವಾಪಸ್ like remember that only. In a room, let's say room of 50 people can join but only 5 people have come. Obviously they can run everywhere, no? So obviously they will not get stained dark. So lightly stained and these people are transcriptionally active. Okay, transcriptionally active. I hope this is clear to all of you. Okay, I hope this is clear to all of you. So in a chromatin there are two regions, heterochromatin and euchromatin. Heterochromatin ಬಂದ್ಬಿಟ್ಟು darkly stained, transcriptionally inactive. Euchromatin ಬಂದ್ಬಿಟ್ಟು lightly stained and active. Okay, on diagram ತೋರಿಸ್ತೀನಿ students. So this is how it looks under the microscope. Okay, these darkly stained regions which you are seeing, no, this ones, these are your heterochromatin. Okay, these are your heterochromatin and in light ag tosta ilva, this is your euchromatin. Okay, illu nodi a border banbitu heterochromatin, darkly stained, a olagirodu euchromatin. Okay, so in a chromatin there are regions where they are darkly stained and there are regions where they are lightly stained. Okay, this is called as euchromatin and heterochromatin. Done, done? Yes or no? Pakka? All of you understood everything? Yes. Please let me know. Done. Pakka. Okay. Done. Okay. Done. Okay. Now, moving forward, students. Search of genetic material. Okay. Ma'am, how did they find this DNA? How did they know DNA is the genetic material? What happened with the transformation? ಈ ತರ ತುಂಬಾ questions ಇರುತ್ತಲ್ವಾ, ಅದೆಲ್ಲ ಆನ್ಸರ್ ಹೇಳ್ತೀನಿ. Search for genetic material ಅಂದ್ರೆ when they were doing experiment, something strike them and they were like, okay, wait, this might be the genetic material. So other the explanation. First ಬಂದ್ಬಿಟ್ಟು students, Frederick Griffith. Okay, so he was, listen to me carefully, very easy experiment. Okay, he was working on Streptococcus pneumoniae. He was working on what? Streptococcus pneumoniae. Okay, Streptococcus pneumoniae in students, the one that causes pneumonia. Okay, causative agent of pneumonia. Okay, causative agent of pneumonia. Correct? Yes or no? Causative agent of pneumonia. Okay. Yes. Okay. Now nodi. Okay. Here, causative agent, understood, ma'am? Causative agent of pneumonia. Now, he took two strains. Okay, there were two strains. Okay, two strains of Streptococcus pneumoniae. One strain was something like this, the rough strain. Okay, and there was one more strain which was the smooth strain. Okay, smooth strain. So students, 2 strains were there. One is called the rough strain. One is called the smooth strain. So idalli nodre ಏನು ಅರ್ಥ ಆಗುತ್ತೆ ನಿಮಗೆ? This one does not have a proper coating. ಅಲ್ವಾ? Proper coating. It is somewhat like, random. ಇದರಲ್ಲಿ ನೋಡಿದ್ರೆ ಸ್ಮೂತ್ ಆಗಿದೆ ಇದ್ರದ್ದು ಬ್ಯಾಕ್ಗ್ರೌಂಡ್ ಅಲ್ವಾ? ಸೊ ಸ್ಮೂತ್ ಮ್ಯೂಕೋಪಾಲಿಸ್ಯಾಕರೈಡ್ ಇದೆ, ಸೊ ಸ್ಮೂತ್ ಆಗಿದೆ. ಸೊ ಒಂದು ಬಂದ್ಬಿಟ್ಟು ರಫ್ ಸ್ಟ್ರೈನ್, ಇನ್ನೊಂದು ಬಂದ್ಬಿಟ್ಟು ಸ್ಮೂತ್ ಸ್ಟ್ರೈನ್, 2 ಸ್ಟ್ರೈನ್ ಇತ್ತು. ಓಕೆ, ಇವಾಗ ನೀವು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಬಗ್ಗೆ ವೆನ್ ಯು ಸ್ಟಡಿ ಯು ವಿಲ್ ನೋ ದಟ್ ಎವ್ರಿಥಿಂಗ್ ಹ್ಯಾಸ್ ಮಲ್ಟಿಪಲ್ ಸ್ಟ್ರೈನ್ಸ್. ಓಕೆ, ಸೊ he was working on smooth strain ಮತ್ತೆ S strain. So he also told that students ಈ smooth strain ಬಂದ್ಬಿಟ್ಟು it is virulent ಅಂತ ಈ ಒಂದು ವರ್ಡ್ ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ NCERT ಅಲ್ಲಿ. ವಿರುಲೆಂಟ್ ಅಂದರೆ ಸಿಂಪಲ್ ವರ್ಡ್ಸ್ ಅಲ್ಲಿ ಡಿಸೀಸ್ ಕಾಸಿಂಗ್. So some people told that this disease causing nature could be because of this mucopolysaccharide, this capsule ಅಂತ ಹೇಳ್ಬೋದು ಈ ಔಟರ್ ಕೋಟಿಂಗ್ಗೆ. ಓಕೆ, ದಿಸ್ ಕ್ಯಾಪ್ಸ್ಯೂಲ್, ಅಯ್ಯೋ ಇದೇನು ಕಲರ್ ಚೇಂಜ್ ಆಗ್ತಾ ಇಲ್ಲ. Students, this capsule could be the reason why they are disease causing, some people told. Okay, ಇದರಲ್ಲಿ ಆ ಥರ ಒಂದು ಕ್ಯಾಪ್ಸ್ಯೂಲು ಇಲ್ವಾ, ಸ್ವಲ್ಪ ಸ್ಮೂತು ಅಲ್ವಾ, ಇಟ್ಸ್ ನಾಟ್ ಸ್ಮೂತ್. ಓಕೆ, ಸೊ ಬಿಕಾಸ್ ಆಫ್ ವಿಚ್ ದಿಸ್ ಪೀಪಲ್ ಆರ್ ನಾನ್ ವಿರುಲೆಂಟ್. These people are non-virulent. That means they are non-disease causing. Okay, they are not disease causing. Okay, understood? ಇದು 2 ಸ್ಟ್ರೈನ್. ಓಕೆ, ಇವಾಗ ವಾಟ್ ಇಸ್ ದ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಅಬೌಟ್? ಓಕೆ, ಅದ್ರ ಬಗ್ಗೆ ಮಾತಾಡೋಣ. ಸಿ, ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಬಂದ್ಬಿಟ್ಟು ಸ್ಟೂಡೆಂಟ್ಸ್, he took a mice. Okay, what did he take? He took mice. Okay, mice, whatever you want to tell. So the experiment was simple. First he took S strain. Okay, he injected into the mice. Okay, obviously the mice died. Ma'am, why mice died anta kelbedi, because ನಾನು ಆಲ್ರೆಡಿ ಹೇಳಿದ್ದೀನಿ this is virulent ಅಂದರೆ disease causing. So disease causing ಅಂದರೆ obviously it is going to kill the mice ಅಲ್ವಾ, mice died. Okay, ಅದಾದಮೇಲೆ he injected R strain. Okay, into the mice. R strain inject ಮಾಡಿದ್ದಾನೆ. So what will happen? The mice is alive. Ma'am, obviously it is going to be alive. Why? Because the R strain is non-virulent ಅಲ್ವಾ, non-virulent. ಇವಾಗ ಗ್ರಿಫಿತ್ ಗಾಟ್ ಅನ್ ಐಡಿಯಾ, ಒಂದು ಚೇಂಜ್ ಮಾಡೋಣ. ಐ ವಿಲ್ ಡೂ ಒನ್ ಥಿಂಗ್, ಐ ವಿಲ್ ಹೀಟ್ ಕಿಲ್ ದ ಎಸ್ ಸ್ಟ್ರೈನ್. ಅಂದರೆ ಮ್ಯಾಕ್ಸಿಮಮ್ ಹೀಟ್ ಅಪ್ಲೈ ಮಾಡಿ ಐ ವಿಲ್ ಹೀಟ್ ಕಿಲ್ ದಿ ಎಸ್ ಸ್ಟ್ರೈನ್. ಓಕೆ, continuously you give heat and you trying to destroy the S strain, and he injected into the mice. Okay, he injected into the mice. What would have happened, students? Can anyone guess and tell me what would have happened? ಸುಮ್ಮನೆ ಗೆಸ್ ಮಾಡಿ ಏನಾಗಿರ್ಬೋದು, ಬೇಗ ಚಾಟ್ ಬಾಕ್ಸಲ್ಲಿ ಸ್ವಲ್ಪ ಹೇಳಿ. ವಾಟ್ ವುಡ್ ಹ್ಯಾವ್ ಹ್ಯಾಪೆಂಡ್? ಕಮ್ ಆನ್, ಕ್ವಿಕ್ಲಿ ಇನ್ ದ ಚಾಟ್ಸ್. ವಾಟ್ ವುಡ್ ಹ್ಯಾವ್ ಹ್ಯಾಪೆಂಡ್? ಫಾಸ್ಟರ್. ವಿಲ್ ದ ಮೈಸ್ ಬಿ ಅಲೈವ್? ವಿಲ್ ದ ಮೈಸ್ ಬಿ ಡೆಡ್? Will the mice be alive? Will the mice be dead? Okay, for you talking, for you people it's not less. Okay, like one this aunties you people are. Come on. Come on, quickly. Okay. Silent heart name is Aishu. Okay. Sanjana Shetty. Heat killed S strain is the mice. So injected into the mice. Students, here the mice was alive. Yake? Because the strain got continuously heating, because of which the nothing got transformed. Last ಏನ್ ಮಾಡಿತ್ತು, ಇದು ಮತ್ತೆ ಇದು ಒಂದು ಕಾಂಬಿನೇಷನ್ ಮಾಡಿತ್ತು. ಓಕೆ, ಸೊ he took heat killed S strain, okay, plus R strain. Okay, heat killed S strain plus R strain. He injected into the mice. Okay, he injected into the mice. ಇವಾಗ ಏನಾಯ್ತು ಸ್ಟೂಡೆಂಟ್ಸ್? ಇವಾಗ ಏನಾಯ್ತು? ಓಕೆ, What happened? Ma'am, yell. Oh. Okay. Thank you, Aishu. Okay. So, if they want to go, let them go. No problem. Okay. So, here the mice was there. Here, students, the mice died. Okay. Mice died. So, Griffith was shocked. Griffith was like, huh? Here I used heat-killed estrane and I injected the mice was alive. ಇದೆರಡು ಕಂಬೈನ್ ಮಾಡೋ ಟೈಮಲ್ಲಿ ಏನೋ ಆಗಿದೆ ಅಂತ. Okay. So, he wrote the conclusion that something from heat-killed estrane, okay, from heat-killed estrane got transformed. What happened, students? Got transformed to the R strain, okay, to the R strain, making, okay, making R strain virulent. Virulent andre disease causing, illandre mice will not die. Okay, so something got transformed from the heat killed strain to the R strain, making the R strain virulent. Okay, making the R strain virulent. Understood? This was Griffith's experiment. This was Griffith's experiment. Clear to all of you? Yes or no? Please let me know. So, conclusion, he did not know, students. He did not know what to call that. So, he called this transformed thing as the transforming principle. What did he call it as? The transforming principle. Okay, let's read from NCERT as well. So, in 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumoniae, bacterium responsible for pneumonia, witnessed a miraculous transformation in bacteria. During the course of his experiment, a living organism had changed in physical form. When Streptococcus pneumoniae or pneumococcus bacteria are grown in a culture plate, some produces smooth, shiny colonies, that is the S, okay, while the other produces rough colonies, okay, rough colonies. This is because S-strain bacteria have a mucus, that is the polysaccharide coat, okay, while the R strain does not have it. Okay, artha aita? Coat anta helilva? R strain, S strain anta. Mice infected with S strain die from pneumonia infection, but the mice infected from the R strain will not die because of pneumonia, because the R strain is non-virulent. Okay, R strain is non-virulent. So, S strain injected into the mice is dying. Okay, because it is S strain. Okay, because it is S strain. Problematic one, yaar students. R S S is problematic. Okay, R strain bandbit students. Obviously, the mice is going to live. Okay. Okay. Sanjana, why you not able to hear? Aishu is her name. Griffith was able to kill bacteria by heating them. He observed that heat-killed S-strain bacteria injected into the mice did not kill them. So, first what did he do? He did heat-killed S-strain and the mice was alive. Okay. He did a combination. He did a combination. Mix madi. He noticed that the mice died. So, suddenly he was like, what would have happened? So, he called that. Students, illinda, see, there was something that transformed, alva. Illinda, illige, eno on transformation agide. So, adanna, he called it as the transforming principle. So, Griffith did not know what got transformed. So, he called that as the transforming principle. Clear? He called it as the transforming principle. Clear to all of you? Yes or no? Okay. Now, injected a mixture of heat kill, I told you, no. So, he called it that some transforming principle transferred from the heat kill S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. So, they say that the, the coat, the mucopolysaccharide coat is the reason for their virulence. Okay, they are becoming virulent with, see here I told capsule alva. So, it is becoming virulent because of the mucopolysaccharide. Okay, mucopolysaccharide. Clear? Yes or no? So, this is your Griffith's experiment. Okay, ee diagram nodi. So rough strain, smooth strain, heat killed S strain and rough strain plus smooth strain. Okay, got it? Yes, done. So this is your transforming principle. So transforming principle, no one knew, students, what got transformed. Okay, no one knew what got transformed. Okay, Griffith called it transforming principle. So Griffith was like, okay, transforming principle. Then another person came into existence. Okay, another person came into existence. No, and another people came and they were like, okay, wait, I really want to know what is the, like, who got transferred. Okay, who got transformed from S strain to R strain. For that, they did the biochemical characteristics of transforming principle. How did they do? I'll show you. Wait. 1, 2, okay, 3. Try to understand. Okay. So, you'll notice, students, simple. Okay. So, first, avaru enzymes etkondidare. ಏನು etkondidare? Enzymes. Okay. And who are the people who did the biochemical characteristics? Avery, McLeod and McCarty. So, biochemical nature of your transforming principle was taken care by Avery, McLeod and McCarty. So, what were they thinking? They were like, okay, yeno transform agide from the heat killed S strain to R strain. We want to know what is the characteristic of that. What would have been inside that killed the mice or that made the R strain virulent. S strain was the villain. R strain was the power fellow. Now suddenly when we mixed these two people, suddenly R strain became the villain and killed the mice. So they wanted to know what happened in R strain that made R strain the villain. So other do biochemical characteristics of the transforming principle. So adukku avaru ಏನು ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ, 3 enzymes ನ ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ. Because ಅವ್ರಿಗೆ ಡೌಟ್ ಇತ್ತು, RNA ಇರ್ಬೋದಾ, DNA ಇರ್ಬೋದಾ, ಇಲ್ಲ ಪ್ರೋಟೀನ್ ಇರ್ಬೋದಾ, ಇದಲ್ಲಿ ಮೂರಲ್ಲಿ ಏನಾದರೂ ಒಂದು ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ ಆಗಿರ್ಬೋದು. So they took all the 3 enzymes that will degrade these people. Okay, so enzymes, they took, students, protease, they took RNAs, they took DNAs. Okay, DNAs. So they took 3 of these enzymes. Now understand this, what is the meaning of this A's, you know? These people are enzymes that will degrade. Andre, listen to me. Protease will degrade protein, RNAs will degrade RNA, DNAs will degrade DNA. Understood? So they took this 3. Okay. So let's take example, protein. Okay, green in color. All the protein is going to be green in color. Okay. Now blue color is going to be RNA. Kansta idea, students? Students, energy tumba kammi agide, students. Nanik demotivation aagta ide. Okay, if your energy is less, even my energy will be less because I am talking to the camera. You should give me the energy. Okay, you should give me the energy how you gave yesterday RCB when they were playing the match in some stadium and you were sitting in the TV and watching. So 3 sets, okay, biochemical nature they were trying to take. First, startingly they use the protease. So what will protease do? Ma'am, it will degrade the protein. So if they degrade the protein and if the mice is alive, namak artha agutte, okay, protein is the transforming principle, alva. So when they use the protease, students, that time also they noticed one thing. Idella biochemical nature ellinda etkondidare from that combination heat killed S strain plus R strain combination ilva. They took this combination's biochemical characteristics. Okay, so they first took protease. Protein got degraded, but still what happened? The mice died. Why mice died? Ma'am, because obviously, ma'am, DNA is still active. So they understood that, okay, protein is not the material. Okay, now again they went to the next step. They took RNAs. Will the mice be alive or dead? All of you tell me, will the mice be alive or dead? Quickly in the chat box. Come on. Tell me. Yes. So here also the mice is going to die. Why? Because here also DNA is still active. So proteinu alla, students, proteinu cancel aito, vital cancel martini. Okay. ಪ್ರೋಟೀನು ಕ್ಯಾನ್ಸಲ್ ಆಯಿತು, RNA ಕ್ಯಾನ್ಸಲ್ ಆಯಿತು. ಓಕೆ, ಲಾಸ್ಟ್ ಯಾರಿರೋದು DNAs ಅಲ್ವಾ. ಸೊ ವಿ ವಿಲ್ ಯೂಸ್ DNAs ಆ್ಯಂಡ್ ವಿ ವಿಲ್ ಡಿಗ್ರೇಡ್ ದ DNA. ಓಕೆ, ಅಂದರೆ DNA ಆ್ಯಕ್ಟಿವ್ ಆಗಿರಲ್ಲ. ಸೊ ಇಲ್ಲಿ ನೋಡೋ ಟೈಮಲ್ಲಿ ದೇ ಅಂಡರ್ಸ್ಟುಡ್ ಒನ್ ಥಿಂಗ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ದ ಮೈಸ್ was alive. So by this what do we understand? That if the DNA is not present, the mice would have been alive. That means DNA got transformed from the heat killed S strain to the R strain. Okay, ide experiment, Avery, McLeod and McCarthy's experiment. But ಈ ಎಕ್ಸ್ಪೆರಿಮೆಂಟಿಗೆ ಒಂದು ಪ್ರಾಪರ್ ಕನ್ಕ್ಲೂಷನ್ ಇಲ್ಲ. ಈ ಎಕ್ಸ್ಪೆರಿಮೆಂಟು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡೋಕ್ಕಿಂತ ಅವರು ಕನ್ಫ್ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ. ಆ್ಯಂಡ್ ಮೆನಿ ಪೀಪಲ್ ಡಿಡ್ ನಾಟ್ ಅಗ್ರಿ. ಓಕೆ, ನಾಟ್ ಲೈಕ್ ಮೀ ಆ್ಯಂಡ್ ಯು, ಲೈಕ್ ದ zoologist and botanist did not agree to this experiment, so they were rejected. But they took almost 10 years to complete this experiment and give this conclusion, but still it was rejected. Okay, it was not considered as the best experiment to prove DNA is the genetic material. Okay, DNA is the genetic material. So what happened? Let's see here. Okay, so prior to the work of Avery, McLeod and McCarthy, the genetic material was thought to be a protein. So people thought that the genetic material was protein. Okay, they didn't know students. DNA and the namagiva gotto. Avarga time al gottidilla. Okay, they worked to determine the biochemical nature of the transforming principle in Griffith's experiment. They purified biochemicals, that is going to be protein, RNA, DNA, etc. Okay, what did they do? One second, yeah. They purified biochemicals from the heat killed S cells to see which one could transform live R cell. Okay, so they extracted it from the heat killed S cells and they wanted to see the transformation in the R cell. Okay, now they discovered that DNA alone from the S bacteria caused the R to be transformed. They also discovered that protein digesting enzymes, protease, okay, protein digesting enzyme protease and RNA digesting enzyme RNase, okay, RNA digesting enzyme RNase did not affect the transformation. So the transforming substance was not a protein or an RNA. Digestion with DNAs did inhibit the transformation, suggesting that DNA caused the transformation. Clear? Simple? Okay, they conducted DNA is the hereditary material, but not all biologists were convinced. Okay, not all biologists were convinced. Clear to all of you? Yes, experiment is clear. Okay, last topic for the day. Okay, Hershey and Chase experiment. After this, the entire search for genetic material complete agutte. Okay, search for genetic material complete agutte. Okay, come on. Hershey and Chase experimentally first namage setup nodana, what and all they took anta. Okay, first they took a bacteriophage. Okay, they took a bacteriophage. Okay, inside this bacteriophage genetic material will be there. We should always draw it with a different color. Okay, yeah. So what is this? A bacteriophage. What is a bacteriophage? A virus that can infect bacteria. Okay, a virus that can infect bacteria. Okay, virus that can infect bacteria. First equipment, okay, they have taken. Now second equipment our ಏನು ಎತ್ಕೊಂಡಿದ್ದಾರೆ students, they have taken S35, okay, radioactive, okay, radioactive substance to label, okay, to label the protein, and they have taken P32, again radioactive, to label the DNA. Okay, label the DNA. This is what they have taken. Okay, yes, this is what they have taken. Now other than this, they have taken the bacteria, which is your E. coli. Okay, E. coli bacteria they have taken. Clear? E. coli bacteria they have taken. This is the equipments that they have used. Now ಈ equipments ಇಂದ ಏನು ಮಾಡ್ತಾರೆ ಅಂತ ನೋಡೋಣ. ಫಸ್ಟ್ ನಾವು ಇದನ್ನ I will explain you this one. Students, sulfur, that is S35, is only present in protein. Only present in protein. So will they affect the DNA? No ma'am, they are not going to affect the DNA. If they are only present in protein, how will they affect the DNA? So they are going to help me in labeling the protein, okay, because they are radioactively labeled. So they are going to help me label, okay, label the protein means what, students? This is labeling, okay. Okay, this is the labeling and my DNA is going to remain normal, okay. So now it is protein is radioactively labeled. Now on the other side, I have P32 who is only present in DNA, only in DNA, not in protein. So will they affect the protein? Of course not, they are not going to affect the protein. So here what is going to happen? This is how the labeling is going to happen. We have labeled only the genetic material. Understood? Now we will see what they exactly did in the experiment. Okay, let's see what they have done in the experiment. So here you see, students, how they have done the red color. So il protein capsule radioactively label agide, il DNA radioactively label agide. So they are trying to see who is going to enter into the cell, who is going to do the transformation. So first step banbid infection. Infection alli ಏನು ಮಾಡ್ತಾರೆ? Obviously the E. coli cell will be infected. E. coli cell will be infected with the bacteriophage. So remember that any virus, okay, any virus when they affect our body, first thing they do is they will release their genetic material. So illu nodi students, avaru first ade ಮಾಡ್ತಾ ಇದ್ದಾರೆ, they are releasing the genetic material, okay, they are releasing the genetic material into the cell. Okay, if you want neat notes also, you can follow me on my telegram channel Gopika Biology official. So telegram alli hogi nanna hesaru amele nanna subject haakidre you will be able to find my telegram channel. Instagram alli follow madbekandre Vedantu Karnataka Instagram channel ide. With that you also have Gopika Biology Instagram channel, so you can follow me there also. Okay, if you want to talk to me or something, you can do that. Okay, now look here, genetic material. So here what is going to happen? Okay. Okay, see here blending they are going to do. What is blending? Where they are going to remove the protein code. With one grind they will be able to remove the protein code. Okay, last one bit centrifugation. Centrifugation andre at a certain speed we will be rotating the whole tube so that we get the heavier particle and the lighter particle here. So here nodi students, no radioactivity detected in the cell. Andre DNA has not entered the cell, like DNA has entered the cell but they are not labeled. So namak correct ag nodre radioactivity detected in the supernatant. Now what is supernatant and pellet? So this is your entire centrifugation tube. Okay, all of you in your smaller age, have you played with mud and water? Okay, you would have played mud and water, you would have made sambar with that and all of that. So there you would have noticed that after some time you will notice that the mud gets settled in the bottom and the water will remain on the top. Okay, water will remain on the top, correct? So this water becomes the supernatant. So what did they see in the experiment? They noticed that protein is present here and where the pellet is there, okay, where the pellet is there, the pellet has the DNA, the genetic material, okay, genetic material. So radioactivity detected in the cell, no radioactivity detected in the supernatant. So understood this? Yes or no? So here you will notice that inside the cell there was radioactivity indicating that DNA is the one who got transformed inside the cell, okay, inside the cell. So this is your entire thing. Now the uni-equal proof, andre students, like the one and only, okay, proof that DNA is the genetic material came from the experiment of Alfred Hershey and Martha Chase in 1952. They worked with virus that infects bacteria called bacteriophages. The bacteriophages attacks to the bacteria and its genetic material, then enters the bacterial cell. So sorry. Okay, yeah. So the bacteriophage attaches to the bacteria and its genetic material and then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it is its own, okay, and subsequently manufactures more viruses particles. Hershey and Chase worked to discover whether it was a protein or a DNA from the viruses that entered the bacteria. ಅವ್ರಿಗೆ RNA ಬಗ್ಗೆ ಡೌಟೇ ಇದ್ದಿಲ್ಲ. ಅವ್ರ ಡೈರೆಕ್ಟ್ ಡೌಟ್ ಏನಿತ್ತು, DNA or protein ಇದಲ್ಲಿ ಏನು, what among this would have entered into the cell. Okay, yes. Now they grew some viruses on a medium that contained radioactive phosphorus and some other on a medium that contained radioactive sulfur. So you'll notice, students, radioactive phosphorus and radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA, but but not radioactive protein. I already told you this. Because DNA contains phosphorus but protein does not. Similarly, viruses grown on radioactive sulfur, same alva, vice versa. Radioactive sulfur did not have radioactive DNA. Okay, no radioactive DNA because DNA does not contain sulfur. Okay, clear? Okay, now here, radioactive phages were allowed to attach to E. coli bacteria. Then as the infection proceeded, the viral coats were removed from the bacteria by agitating them in the blender. The virus particle was separated from the bacteria by spinning them in the centrifuge. Bacteria which was infected with virus that had radioactive DNA were radioactive, indicating that DNA was the material. Okay, so the one, student, simple agide. The ones where DNA entered into the cell, DNA was already radioactively labeled, so obviously cell radioactive agide. So from this, what do we understand? Yes, DNA is the one that gets transformed. Okay, the virus to the bacteria. Bacteria that were infected with virus that had radioactive protein were not radioactive. Okay, radioactive protein were not radioactive, but DNA was the material that passed, because of which the cell was radioactive. Okay, this indicates that proteins did not enter the bacteria from the virus. DNA is therefore the genetic material that passed from the virus to the bacteria. Clear? Yes or no? Understood? All of you understood? Okay, understood. Okay, this is the story. Okay, now students, who and all did not join in the starting, nan solpa informations helidini. Those students who are looking for NEET, JEE matte KCET idu what is it, coaching, ನಿಮಗೆ JP ನಗರ್ ಬ್ಯಾಂಗ್ಳೂರಲ್ಲಿ ಒಂದು ವೇದಾಂತು ಲರ್ನಿಂಗ್ ಸೆಂಟರ್ ಇದೆ. ಸೊ ವೇದಾಂತು ಲರ್ನಿಂಗ್ ಸೆಂಟರಲ್ಲಿ ಹೋಗ್ಬಿಟ್ಟು ನಿಮಗೊಂದು ವಿಸಿಟ್ ಬುಕ್ ಮಾಡ್ಬೋದು. ಲಿಂಕ್ ಡಿಸ್ಕ್ರಿಪ್ಷನಲ್ಲಿ ಇರುತ್ತೆ. ಸೊ ಬುಕ್ ವಿಸಿಟ್, ಆ ಬುಕ್ ವಿಸಿಟಲ್ಲಿ ನಿಮಗೆ ಅಲ್ಲೋಗಿ ಅದರದ್ದು ಫೆಸಿಲಿಟೀಸ್ ಹೆಂಗಿದೆ, ಕ್ಲಾಸಸ್, ಡೆಮೋ ಕ್ಲಾಸಸ್ ಎಲ್ಲ ನೋಡ್ಬೋದು. ಅದಾದಮೇಲೆ ಅಲ್ಲಿ ಜಾಯಿನ್ ಮಾಡ್ಬೋದು. ಸೊ ಇಟ್ ಇಸ್ ಇನ್ JP ನಗರ್ ಬ್ಯಾಂಗ್ಳೂರ್. ಓಕೆ, JP ನಗರ್ ಬ್ಯಾಂಗ್ಳೂರ್. Very nice area, JP Nagar, Bangalore. So JP Nagar, if you are any of you are staying near JP Nagar, who can travel, who can commute, you can go to JP Nagar, book a visit. First you visit, you see everything, then only you can join if you are a JE, NEET or KCET aspirant. Okay, clear? And students, yella notes ಬಂದ್ಬಿಟ್ಟು Vedantu Karnataka telegram channel ಮತ್ತೆ ನನ್ನ telegram channel ಅಲ್ಲೂ ಇರುತ್ತೆ. So ಖಂಡಿತ join ಮಾಡಿ telegram channel. Okay, yes, cleared all of you? So students, naale 2 PM class ikkidini because I have another class also after that. So tomorrow the same part 2 will be at 2 PM. Okay, amele after that you will be starting your exam 3's classes. Okay, exam 3's classes will start, maths also will start soon. So exam 3's classes will be there. So those who know any friends who have not cleared exam 3, please share this channel with them. It is going to come very helpful for all of you. Okay, yes, okay. Thank you so much, students. So if you are new to this channel, do not forget to subscribe to the channel, and if you like this class, please let me know in the comments and quickly smash the like button. This class is a breakdown of your entire NCERT plus my handwritten notes. These notes will be shared on the Telegram channel. I have made better notes before also, so you can take it from my Telegram channel as well. Okay, thank you so much, students. Take care. Bye bye. I hope all of you enjoyed the class and understood every single concept. Each chapter, one of the most important chapters of class 12. NEET gu tumba high weightage chapter, KCET gu tumba high weightage, board exam go high weightage chapter. So if you're a student who's preparing for any of this, do not miss this chapter at any cost. Okay, so thank you so much. Bye bye. Thank you guys. Bye bye.